Good morning. How wonderful to see you all here at the annual holiday lecture series. This was originated over 52 years ago by one of our late and fantastic professors. I'm glad to see the turnout. It's fortunately not with 30 inches of snow, which it sometimes is. I'm introducing a very special lecture highlighting and marking the incredible work of a very close friend, Ralph Steinman. Ralph Steinman and I knew each other for over 40 years here at the Rockefeller University. Ralph and I are both physician scientists and professors and heads of laboratories. You're going to hear a great deal about the phenomenal work of his laboratory, and I'll simply say my laboratory bears the title it always has, the Laboratory of the Biology of Addictive Diseases. On October 3, while I was on a plane to Stockholm, two things were announced. Ralph had just won the Nobel Prize, the greatest prize awarded to a scientist. International prize, coveted by all, won by a very few, and very deservedly. I also learned that he had passed away 3 days before. It was a bit of a tough ride, but I went with the pride and the love of Ralph and his family and all of his workers. Today's lectures will highlight the work that was awarded the Nobel Prize. That is, Ralph's work in the discovery of dendritic cells, a very special cell type, which he found and which he went on to study for the development of medications and vaccines to treat a variety of disorders and protect against them, including HIV, AIDS, and a variety of forms of cancer. Today there'll be two lectures, one in the morning and one in the afternoon, from two leaders from Ralph's laboratory. We're going to hear how he first discovered the cells in 1972 by simply looking into a microscope. How many of you have looked into a microscope? Well, keep your eyes open when you look into the microscope. You may see something you didn't expect to see, and don't think you're wrong. Just keep looking at it, because what Ralph found was very unusual. And he realized what these cells were, which is different, and then defined what they were, which is very special. It's a wonderful story, and it will be written for years to come as scientists around the world are working based on his findings to make medications and vaccines for a variety of diseases. Slide. Jim, can you progress? I can't progress. It's not progressing. Oh, there's progressing. Good. So this is 1992. We had known each other for about 20 years at that time. And this is the picture, the annual picture of the hospital. And it was a windy day, but Ralph and I frequently would stand together in pictures because we remember from the 70s to really the mid-80s, no one believed that he had really found a new kind of cell. And no one really believed all the wonderful things it did. It was slow, it was coming, but bit by bit, different wonderful immunologists learned. No one believed that I had really defined that addictions, alcoholism, cocaine addiction, nicotine addiction, are diseases of the brain and not some personality trait disorder. And that medications coupled with behavioral care can make people well. And we have shown this now with 1 million people in methadone maintenance treatment worldwide, which we developed here. But we kind of bonded closely because we were doing different things and heading different laboratory groups. Ralph was ultimately not only believed, but followed by the majority of immunologists in the world, if not all, and many of us beyond. Although he is no longer with us, his legacy will go on forever. Slide, please. Sarah Schlesinger, I will brag, she went to the same college I went to, a tiny college, a women's college, Wellesley. And she went on, as I did, to go to medical school at Rush. And then she went on to be chief resident in pathology across the street at New York Cornell, New York Presbyterian Weill Cornell, called today. And then she went down and worked with the Armed Forces Institute of Pathology, and she worked on developing an AIDS vaccine. She was at Walter Reed in its original site, and now I'm sure visits at its beautiful new site. And she worked both in Bethesda and in Washington, but around the world, with her development of AIDS vaccine. And then, having worked here at Rockefeller from the age 17 onward while a student, hint, hint, I worked at the NIH while a student in high school and college. She then returned here to Ralph's lab in 2003 and has played a marvelous role with Ralph as a friend, a colleague, and heading up his clinical research vaccine team. Slide, please. Marina Kasky is yet a new generation of physician scientists. And she was a clinical scholar here after having graduated from a university in Brazil. She went to the Federal University of Sergipe, I hope I pronounced that almost right, and then came to New York to do her house staff residency at St. Luke's Roosevelt, which is affiliated with Columbia PNS. And then she came to be a clinical scholar here at Rockefeller, a fantastic program connected with our hospital here and our clinic, where Ralph and I did, and I still, active present, do very, very constant and exciting research. And she has joined the laboratory of Ralph. Sarah is an associate professor of clinical investigation, and Marina is very recently now an assistant professor of clinical investigation. So we have a litany of women as well as men at this university doing science, and in our cases, as physician scientists. I would also like to acknowledge Ralph's family. This is Claudia, his wife, in the front row, receiving the Nobel Prize on December 10 from the King of Sweden at Town Hall, a beautiful, beautiful place. And she is receiving it. I hope you'll get to see some more of the pictures another time, with much of the family in attendance. And Michel Nussensweig, one of his most outstanding trainees, had given the Nobel lecture just before, on a previous day. So this is the story of the Rockefeller University, our connectivity with each other, our closeness, our family here at the Rockefeller University. And with no further ado, I'm going to turn the lecture over to one of my close friends and colleagues, Sarah Schlesinger.
So good morning, everybody. I am delighted to be here, and I'm delighted that you're here. So we are here for an hour and a half now. We'll have lunch, which is pizza, for those of you who like to look forward to lunch. And then Marina will take the podium in the afternoon to tell you the rest of the story. So as Mary Jean told you, I'm going to tell you about Ralph's amazing discovery of dendritic cells, and then Marina is going to tell you about some of our more recent work, which is more clinically applied. But we're going to be sitting here for a long time, and I'm so delighted to see so many of you taking a day of your precious Christmas vacation, or holiday vacation, to join us to explore science. But you guys can't sit there or you'll fall asleep and you'll miss the fun. So what I'd like for you to ask questions. There are 2 microphones, I think. Where are the microphones? Over there and over there. This is webcast, so even if I can hear you, people who are watching on the webcast won't be able to hear you unless you use one of the microphones. So at any time if you have a question, please ask it. I will be asking you questions, and there is an incentive for both asking and answering questions, and that is my little Carol Merrill moment. These are little stuffed dendritic cells, And these are pathogens, okay? A whole bunch of different germs, each one cooler than the next. So if you ask or answer a question, you can come and get one of these. I would throw one to you, but I've got a really bad throwing arm, and it probably wouldn't get to you. So these are really cool. And my favorite one of these, that's the Black Plague. Thank you. This is a superbug, okay? This is Staphylococcus aureus, which grows in clusters, and it's a superbug because it has a superhero cape. So if I asked a good question, that's the one I'd go for. So feel free at any time to interrupt and ask a question, and you too can have a superbug. So, as Mary Jean told you, we are here today for a remarkable reason, and that is to celebrate and to learn about the work that led to the awarding of the 2011 Nobel Prize in Physiology or Medicine. And the citation for the prize read, for his discovery of the dendritic cell and its role in adaptive immunity. And I put here, this is what the Nobel Award actually looks like. And also, as Mary Jean told you, sadly, Ralph died 3 days before he was able to even learn that he'd received the prize, and he certainly wasn't able to attend the ceremony in Sweden on December 10th. But his lovely wife, Claudia, received the prize in his stead, and here you see her receiving it right there from the King of Sweden. And Claudia is here today with 2 of Ralph's children and 2 of his granddaughters. Ladies, could you come up and show everybody? Come. So this is, turn around so people can see you girls. So this is, you want to say your name, or should I say it?
Hi, Nisla.
And who's this?
Sila.
This is Sila. And they are getting ready, like you guys, to be scientists. So they're here too, and they'll help us through some of the stuff, okay? All right, you want to go back and sit down? Okay. Now, I am here, as Mary Jean told you, because I've worked with Ralph for virtually all of my professional life in one way or another. And my professional life started when I was 17, even though I didn't realize it at the time. But I'm uniquely qualified to be giving this lecture because 35 years ago, I was in the audience. So I am not a hoarder, but I managed to keep both this little letter, and I'll show you my ticket, that I was sent 35 years ago. And this only kind of shows how things have changed. The stationery is a little simpler. We have a different zip code. And this was actually typed on a typewriter and signed by hand. And if it says here, Ms. Lewis says, feel free to call me at this number with no area code between the hours of 9 and 2, Monday through Friday, because that was the only way to reach her. There was no cell phone, and if you called, you wouldn't get voicemail. So things have changed a bit. These were also the Alfred E. Mirsky Christmas lectures on science as opposed to the holiday lectures. And I just wanted to say a word about Alfred Mirsky, for whom the lectures were named. He was a scientist here at the Rockefeller who believed you got people into science by hooking them early. And I think it works. It worked for me, and I hope it will work for you. So this was my ticket. They were perforated tickets then. They only took the first lecture, and there were 2 days of lectures, and they were only in the afternoon. But I had the great pleasure, and really it was a life-changing experience for me to hear Dr. Christian de Duve lecture on a guided tour of the living cell. This was 1976, and he had won the Nobel Prize in 1974 for his work describing the microanatomy of the cell with George Pilati. And you'll hear a little bit more about that later. But it was a remarkable and inspiring experience for me, not just to hear about his discoveries, but to hear about how he did it. And so that's what we're going to try to tell you today, to teach you a little bit about what Ralph discovered, but perhaps more importantly, how he did it, because that's really the fun and most interesting part. Oh, so this is Dr. de Duve, and he was, so in 1974, he was at the peak of his sort of academic powers, and he's in his mid-90s. I think he's 95 or 96 now, and he's still working. And I had the great privilege of sitting with him. Was one of my good friend and colleagues here, Jackie Chiapetta. Her sister, Adelaide, works with him and arranged for me to meet him. And the lecture that he gave is actually published in a wonderful Scientific American book. And he gave it to me, which I didn't even know. So if you are interested, you can too have a guided tour through the cell. And why? Okay. And this is my ID card from 1979. I couldn't find the one from 77 or 78, but we had to blur out the Social Security number, which appeared on it. It was laminated. It had no electronic capabilities. My ID card now gets me into all sorts of doors and stuff, but it did have my picture, which I don't look like anymore. So, and this is the last of my reminiscences. This is a picture of our lab from 1980 to 81. And this picture actually hangs on the wall currently. And I don't know if you can find me or find Ralph, but we'll help you do that. So here I am, again, looking quite a bit different. And there Ralph is, looking pretty much the way he always did. So I got to work for Ralph through a series of very unusual, totally random coincidences. I came home from the lecture and told my parents, we had never heard of this institution. I don't know how many of you had heard of the Rockefeller before you came here, but I hadn't. And I said it looked like it would be a really good place to have a summer job. So my parents said, that's fine with us if you want to. And my mother knew somebody who knew Claudia, Ralph's wife. And I got Ralph's phone number and I just called him. Now, fortunately or unfortunately, things are more organized now. And I don't suggest that you randomly call or email people. But we have a program for high school students who are interested, and it's called the Rockefeller University Science Research Program, or the Outreach Program. And if you go to our website and Google, you'll find it. And there's an online application, and I highly recommend the program. It's run by Ted Scoville, and it's a terrific way to come here and really learn science by doing science. So this is Ralph on his first day of physical diagnosis at Harvard Medical School. I guess he did look different then, and that was 1964. So Ralph, as Mary Jean mentioned, we are all physician scientists. So there are people who have MDs and there are people who have PhDs, and we are scientists with, as I like to say, only an MD degree. Both paths are good. Both paths give you lots of opportunity. But being a physician scientist is a little bit different. And Ralph spent most of his life in the beginning working as a basic scientist. And then in the last decade or so, he took his discoveries and he applied them to medically important problems in a more directed way. So you're going to hear about both phases of his career. And in Ralph's office, he had on a whiteboard, and you know a whiteboard that you write on, and on that whiteboard were many quotations. And so obviously, and it would be silly of me not to mention this, we would all prefer that he were here to give this lecture. You would prefer to be hearing it from a Nobel Prize winner himself. We all wish he were here. And believe me most of all, would rather be sitting in the audience and listening than being up here, though it is obviously a great honor and a great privilege for me to be here. But I want to give you, as best I can, some sense of Ralph, some sense of his enthusiasm, his pleasure in science, and how much fun he thought it was. And so throughout the lecture are a few little quotes, some of which he had up on his whiteboard, and some of which he sent me in an email when I asked him for this one to include in a lecture I was giving. Also, he had a way of collecting objects that looked like dendritic cells. So they had nothing to do with them, but they just reminded him. So you're going to see images of some of those objects through the talk as well. So my French is really crummy and my accent is horrible. It was always a great comfort because even though Ralph's French was fluent, his accent was worse. He'd grown up in French-speaking Francophone Canada. So I'm just going to go for the English, which is chance favors the prepared mind. But this is the Louis Pasteur quote, and he gave it in a lecture in 1854. So I think that there's a lot of truth to that, and it applied to Ralph. In the end, any discovery hinges on a certain amount of luck and a certain amount of magic. But he was extremely well prepared. And that's what you can do. You can prepare yourself. You can train yourself. And then when it happens, you can avail yourself of the luck. So this is one of the images that I'm going to show you. And these are glass objects that were made, if you are interested, in Venice by a Mr. Galati, who never knew about dendritic cells, but they've become dendritic cells for us.
Science became important for me when I was in college. When I entered college, I really didn't have a focus in terms of what I was interested in professionally. But that is when I took my first course in biology, and then I was hooked. Nothing else mattered.
When did you first sort of set your sights on looking at dendritic cells?
When I began my postdoctoral research, what I was really trying to do was understand a problem in immunology and medicine. And the problem was, how do you start your immune reaction? So the immune system is very important in many diseases, but we didn't understand how the whole thing gets going. So if there's an infection and your immune system has to respond, how do those two come together? How is the response initiated? And we were looking in a very simple, contrived experimental system, which was the only one I could identify to study this problem. And we found this totally new cell. I couldn't have anticipated it. It was brand new. But we were so sure this was a new entity that it was really relatively straightforward to persevere.
Now, everything he said was absolutely true except the very last thing. It wasn't relatively straightforward to persevere. And one of the things that made Ralph so different from most other people, most other scientists, was his perseverance, his willingness to stick with what his observations were. So as Mary Jean Creek told you, and is actually in the title of this was a very controversial discovery. And he discovered the cells in 1972, and it really took 10 to 15 years before they were accepted by the rest of the community. And during that time, Ralph worked really as probably the only person, or his group worked as the only group working on dendritic cells. And it was straightforward for him, because once he saw it and he knew it, that was it. But for the rest of the world, it wasn't so straightforward. So now I'll catch up. Our cell. And so to understand what Ralph's discovery meant and how he discovered the cells that he did, you need to know a little bit about immunology and what was understood at the time. So immunology really began as a discipline in 1908. It was recognized as a discipline in 1908, with the Nobel Prize being awarded to Paul Ehrlich for adaptive immunity, and he discovered antibody. He called it side chains, but he discovered the idea of antibody. And we've provided for you a handout that kind of goes over all of this, so I don't think you have to take too many notes, or any at all, because you have it all there. So you certainly don't have to take notes, but anyway. And this is Ilya Menshnikov, who discovered innate immunity. He discovered phagocytes. So the cells that we work on are part of the family of phagocytes, and they're cells that eat up other cells or other debris. And dendritic cells link the two parts of the immune system, the innate, which happens immediately, and the adaptive, which takes a little longer and is characterized by memory. So in Metchnikov's Nobel lecture, he said there is no need to be a doctor or a scientist to wonder why the human body is capable of resisting so many harmful agents in the course of everyday life. So if you think about it, we live in a world full of danger, a world full of foreign material, and how is it that with all of those pathogens over there, that we all manage? So it was understood from very early on that people who were infected by the plague of Athens, who recovered, didn't get it again. So the idea that natural infection would protect you from subsequent infection goes back to the ancient world. But nobody understood or had any reasonable approach to understanding how that happened. Jenner, who you'll hear more about from Marina in the afternoon lecture, made observations about milkmaids who were protected from smallpox by a different infection, by cowpox. And he capitalized on that observation to make smallpox vaccine, which is actually the only vaccine that's responsible for eradicating a pathogen from human disease. So smallpox vaccine has successfully eradicated smallpox. But again, he didn't understand why. So those observations are pretty clear. And here we have a person in all of the various pathogens that you're encountering, or one it's encountering every day. And it's our immune system that protects us. So this is, I love this. This is our forgive me. This is our immune system, okay? So you have different cells. You have phagocytes, you have lymphocytes, you have granulocytes, all of them working hard to protect you. We defend. So the essential elements in immunity, and forgive me, for those of you who know this already, just bear with me, because we need to make sure that everybody's on the same page. If anybody has any questions and it's not clear, please ask. So the essential elements in immunity, the first is an antigen. So an antigen is any substance, either foreign or self, that is recognized by the immune system. So that could be any of the microbes, pollen, for an allergy, or vaccines when we intentionally manipulate the immune system. And Ehrlich described side chains, which have subsequently been we learned are antibody, and that's a protein in the blood that's made by B-cell lymphocytes, and this is known as the humoral arm of the immune system. Sometimes these are called antisera. Cellular immunity, which is what we work on, are this direct response of cells in the immune system that directly respond to an antigen. Okay. So this is another little cartoon of my friends here showing you how the immune system works. And the key thing to remember about the immune system are these properties. The first is it's specific. So you make a specific response to a specific microbe or a specific allergen. It's not general. So if you respond to the common cold, that doesn't cross to chickenpox. Then the next property is it's diverse. You make many, many responses, hundreds or thousands, to any given antigen. And perhaps most importantly is that the immune system is characterized by memory. So this is sometimes called amnestic. And what that means is when the immune system learns, when you first encounter an immunogen or a pathogen, you make a response, and ever after that, the immune system remembers and mounts a faster, quicker, more intense response upon re-challenge. So specificity, diversity, and memory. And the constituents of the immune system, and these are the constituents as they were understood in the early 70s, and this remains correct. I'm not going to give you any misinformation, and where things diverge from what was understood, I'm going to tell you. So these are B-cell lymphocytes, and remember I told you they're the ones that produce antibody, or the humoral arm of the immune system. And the B, people sometimes think it stands for bone marrow because they come from the bone marrow, but these were first described in birds. And birds have a common immune organ called the bursa, or the bursa of Fabricius, and they're actually called B-cell lymphocytes for that origin. And they produced immunoglobulin or antibody in response to antigen. And this is a cartoon of the B cells. And so you have antigen, a young or immature B cell, and it receives help, and this is very important, from an activated T cell, which you'll hear about in a moment. It grows up to be a plasma cell, and it pours out, as Ralph would say, buckets and buckets of antibody. And you can see here, and you're going to see better in a moment, the shape of the antibody is a Y shape. So antibody or immunoglobulin is produced by B cells, and it may be neutralizing, which means it kills a bacteria or a virus, or non-neutralizing and just binding. And this is what it looks like. Gerald Edelman, who was here in the 70s, was awarded the Nobel Prize for describing this. So you have this portion, which is the variable region, and this is what's different for each antigen, and this is the common region, or the FC region, the constant region, and this remains the same. And here you have a light chain and a heavy chain. And there are two different versions of light chains, and the variability, as I said, is all in this end, or the antibody binding region, sometimes called the FAB region. T-cell lymphocytes, and these were, for us, these are sort of dogma, but these were described in the 60s. So when Ralph was working, the description of T-cell lymphocytes and the characterization of them was relatively new. They're effector cells, and they come from the thymus, hence the T-cell. The thymus is an immune organ that we all have that sits over the heart. And it's the largest it ever is in when you're 2 years old. So it's pretty large when you're born. It grows till you're 2 years old, and then it involutes. So if you know any 2-year-olds, they've got good thymuses. So CD4 cells or T cells are helper cells. Those are the cells that make the help that you saw helping to make antibody. And then CD8 cells are T cells that are killer cells. And they directly kill on contact, and they are critical in antiviral and anti-tumor immunity. So all of the lymphocytes, T cells, B cells, CD4 and CD8, when you look at them in the microscope, there's no way to tell them apart. They look like little bland, round cells. They have different molecules on their surface, which is how we stain them to characterize them. But they're mimics for each other. When this is a resting helper T cell, and this is a resting cytotoxic T cell. When a helper T cell becomes activated, it pours out cytokines, particularly IL-2. And then when a resting cytotoxic T cell becomes activated, it directly kills. And it does so by secreting, again, directly perforin and granzyme. So perforin, as you might guess from the name of it, makes holes in another cell and kills that way. And granzyme is equally toxic.
Can I ask a question?
Sure.
So I realize that the immune system is characterized by memory, but then why do you need to get booster vaccines when you've already gotten a vaccine?
That's a very good question, and the reason is because it's imperfect. So when you get sick, say, let's take smallpox, which is the best example I can think of. If you get natural infection with smallpox, 70% of people who get infected are going to survive, and 30% are going to die. If you're in the lucky 70% who live, you don't need to get boosted ever again. You have lifelong durable immunity. But the vaccine is a mimic of the natural infection, and it isn't quite as good. One of the goals, in fact, in developing vaccines is to make vaccines that only need to be given once, because it's a pain in the neck for you to go back to your doctor to get a booster. But if you're in West Africa and you have to go out into remote villages, it's a matter of life and death whether you can protect people long term. So it's due to the imperfection in our technology and not the imperfection in the immune system, because a good, robust natural infection will give you durable immunity. You get to come down and pick a bug. Yeah. Good question, and thank you for asking.
Can I get the Black Death?
You can get the Black Death. That is the Black Death, one of my very favorites. All righty. So when we're always looking for vaccines that are durable and don't require boosting. And one of the things that you're going to hear about in the afternoon is Ralph had a great idea that we're working on for making vaccines that are more durable. And if you think you need a lot of boosters with the shots that we get now, there's a current AIDS vaccine that's been tried that shows the ability to protect, which is wonderful, but it only lasts for about 6 months. So terrific that it works, but from a practical point of view, if you've got to vaccinate people every 6 months, you don't have a product. You have a science experiment. So this is an ELISpot. So this is a way of measuring remember I told you CD8 cells secrete various substances which kill directly, and you can't see them, but this is a way we detect them in the laboratory using an ELISpot, so an ELISA technology in spots, and each of those spots represents a cell that's making interferon gamma, which is the cytokine that we were looking for. Once again, one of the beautiful Venetian glass dendritic cells. I would have liked to have been able to distribute them, but we didn't have the budget. So when Ralph came, he knew about T cells and B cells and immunity. And as he told you in his little video that I managed to almost miss, that what interested him this is really creepy, was the question of how the immune response gets started. So how do you initiate immunity? And nobody really knew that. There was some sort of vague understanding that there were accessory cells involved, but it wasn't clear. And very often when something isn't clear, it's because we don't know enough. So Ralph came to work with Dr. Zanville Cohen, who was his mentor, who I had the great privilege of working under for many years, who was a wonderful, wonderful man. And if Dr. Cohen had lived, he would have no doubt shared the Nobel Prize with Ralph for the discovery of dendritic cells, because they worked closely together, and it was Zan's insight and support that allowed Ralph to continue working on something for 16 or 18 years when really very few other people but the two of them believed that it was the case. And he was also a really nice man, as was Ralph. This is Dr. Cohen, and at the time Ralph came to the lab, he shared the lab with Dr. Hirsch, and they worked on macrophages, which were the primary phagocytes in which people were interested at the time. And the lab had been headed previously by Dr. René Dubose, who was a soil microbiologist. And Dr. Dubose discovered the first antimicrobial, anti-tuberculous drug in the 30s. And so he was interested in tuberculosis, and tuberculosis grows in macrophages. So Dr. Cohen had come to work with him on the macrophages that were the relevant cell for the pathogen that he worked on. And deep in there, there is a tuberculosis germ. So now, Ralph again.
We turned to a system in which immunologists would take two sources of cells to generate an immune response. On the one hand, they took cells that are called lymphocytes that mediate the immune response, a fact that was discovered by Sir James Gowans in England about 10 years before I started my work. And then they would take another cell population that stuck to a glass or tissue culture surface. And by mixing the two of them together, they then could add an antigen or stimulus and get an immune response. So I decided I would look at these adherent or sticky cells to see what their properties were, to try to understand how you initiate an immune response. And when I looked down the microscope, I saw something that was totally surprising. We turned to a system in which...
Totally surprising. There we go. Okay, and this is the microscope that he looked down. So this is our new laboratory, but Ralph worked, and when you come out here, when you come out the front, if you look to the right, there's a gray building, which is called the Bronx Lab. And actually, when Ralph came here, it was called the South Laboratory. And he worked there for 40 years. Last August, not this past one, but the one before, we moved to the new lab right across the way from here. But this microscope was with Ralph in the beginning, and it went with him everywhere. And this was the scope in which he looked down and saw the cells. And it wasn't just that he saw them, but he recognized them. And this is his handwriting. He labeled it because people tended to take stuff. Still do. And that's, I guess it'll now be preserved for history. So as Ralph said, when he was interested in figuring out how the immune response was started, what generated immunity. And the system that he worked with was to take B cells and T cells and mix them together and add an accessory cell. So the accessory cell was one that stuck to glass. And remember, there were no monoclonal antibodies to characterize cells, so you couldn't say this is a T cell, this is a B cell. Everything was separated by physical properties, and then they were mixed together, and you would look for an antigen-specific antibody response or plaque-forming cells.
I'm sorry, but I have another question.
Don't be sorry. Ask your
It has
question.
to do with the immune system and how it remembers your responses to everything, basically.
Uh-huh.
So if it does remember, then how do we get the cold so often? Is it like a different strain or?
Exactly. So for that, you get a cold virus.
I also had a question, like what, like
Okay.
what
But
makes the cold so different? Like why is it evolving so often? Why
is
aren't
it?
other
Okay.
viruses?
So you're absolutely on the right track. The reason that you can get a cold over and over again is actually twofold. One is a cold refers to a clinical syndrome. So there are many different viruses that can give you something that feels like a cold. But if it's the rhinovirus, which is the actual cold, as opposed to, say, adenovirus or enterovirus, because there are many different viruses that can give you a similar clinical syndrome, it mutates very rapidly. It's the same problem as influenza. So everybody here, I bet, has a robust response to influenza, whether you've been vaccinated or not. But that doesn't mean that you don't need to get the flu shot, because influenza mutates at such a rapid rate that the virus that's circulating now is sufficiently different from the one to which you made an immune response that you won't be protected. So there's what's called homologous protection and heterologous protection. So homologous protection is being protected from the same thing to which you made the response. And I guarantee you, if you got exposed to the same cold virus, you wouldn't get it again. But it's that the virus has mutated sufficiently to no longer be what's called a homologous challenge. You're welcome. You cannot rely on my throwing abilities. They're worse than my AV abilities. Here. Oh, you're welcome. All right. So, oh, it's not? I don't know though if it'll be so do you mind just walk over there, okay? We'll wait. I'll get my microbes ready. Come, if you've got a question, come up. Go. Yeah, but I want to hear it. But just go stand at the microphone so everybody else can hear it too.
Hello? Okay, this one works.
Is that one's working?
Hi.
Okay, great.
So my question lies with actually your present slide. I understand how you were talking about separating the B cells and the T cells based on physical properties. But whoever did this, how did they know to isolate the B and T cells in the first place?
Okay, so they didn't. So that's a very good question because that clarifies what I said, which is wrong. They didn't isolate the B cells and T cells. They were a mixture of lymphocytes. So what they put in were the cells that they could see as lymphocytes. And these are all coming from mouse spleens. And they looked the same. But they had a mixture of B cells and T cells. It's only later that they would separate them. Okay? So for that, I think you get a white cell. Here, no. You can't rely on me to throw. Come all the way down. There, you're welcome.
Oh, these are adorable.
Okay. Next question.
Yeah, my question's pretty short.
That's fine.
What substance did Ralph use to identify these dendritic cells?
What substance? His eyes.
No, like, what did he use to look in the microscope?
But that's really what happened.
No, like I'm asking, what substance did he analyze to locate these cells?
Oh, okay.
Yeah.
So that's a very fair question. He started with mouse spleen cells, and actually I'm going to get to that, and you're going to see them in just a minute. But so all of these cells, the B cells, the T cells, the macrophages, and the dendritic cells, were isolated from the splenocytes of mice. So mice are a very good experimental system because they're like us in a lot of ways. They're not like us in every way. They're not perfect, but they work pretty well. And the spleen, which is an immune organ, which we have, in people there's a lot of connective tissue in it, so it's hard to get the cells out. But in mice, it's basically, there's very little connective tissue and the cells come out easily. So it was a system that a lot of people worked on, including Michelle and Dutton, and it was where Ralph started. So later on, you're going to hear about dendritic cells from human blood and dendritic cells from human skin. But this part is all mouse splenocytes. And we're going to talk about how he separated them. So if you hold on, you'll hear about it. And if it's not clear, come back and ask. But the most important thing that I want you all to understand is he saw the cells. There was no fancy chemistry. There was no fancy PCR. PCR didn't exist. But it was good observation. It was looking to see what was before him and not what he expected to see that led him to this discovery. And if you come away with one lesson, that's what I'd like you to learn. And you can come down and get a white cell or a pathogen, whatever you like. Okay, you can pick. That's my favorite one. That's the superbug. So hold that one up. Okay, so that's Staphylococcus aureus, which grows in clusters. So it's like a little cluster, but it's a superbug, and it has a superhero cape on it. So I think that's the best one.
Sorry, is it all right if I ask 2 questions?
It's all right, as many questions as you have.
All right, so the first question is, I guess, like, you know, when you get your tetanus vaccine, you're immune for 10 years. Does the immunity period vary, like, by how, like, well the vaccine mimics the disease?
So you're not immune for 10 years. Nobody wants to find how long you're immune for, because if they found how long you're immune, you would get infected. What's known is if you're boosted every 10 years, you maintain your immunity. So tetanus is a really good example because you can measure neutralizing antibody. So remember when we talked about antibody back a few slides, there's neutralizing and non-neutralizing. With tetanus, you get neutralizing antibody. So what's known is that, and you have to look in populations, there are people who can probably get a tetanus shot and be protected forever with one shot, but everybody is protected, or virtually everybody, if you re-immunize every 10 years. So that's one of the things that's really interesting about vaccinology, is that you have these issues of cellular immunity, but then there are these big issues of public health. And so even if you don't need to be revaccinated, if your vaccine is good for 20, as long as somebody in the population needs it every 10 years, the recommendation has to be every 10 years. Okay?
And also for vaccines like the HPV cervical cancer type protection, you have to get 3 of those every 6, you have to get 3, one every 6 months, and then after the 3rd shot, you're protected, I guess, for life?
Right. Well, that's actually a very good question. That's a new vaccine. So the HPV vaccine, which is a wonderful vaccine, it was invented by people colleagues of ours who we know and like, and it works really well. And I would strongly recommend to every one of you, male or female in the audience, that you get it, because it's the first vaccine that protects against cancer. And it protects against cancer because the HPV, which is a virus, the human papillomavirus, actually causes cancer. So it's a pretty amazing thing. With that said, the recommendation is that you get 3 shots, and people appear to be protected for as long as it's been tested. But what will happen is people will continue, the scientists who develop it will continue to monitor those who got it. And if the immunity seems to wane, then recommendations will be issued for people to be boosted. But at this point, it appears that those first 3, the primary series is what it's called, is sufficient to induce immunity. Okay? But again, it's all, come on down, because it's all a matter of continuing to monitor people and scientists making observations. It's not as exact as people would like to think. You get to pick. While she's picking, why don't you go ahead and ask your question?
Okay, so my question is, if it was such a simple, if it was just a cell, then why hadn't anyone seen it before?
So that is the $64,000 question. And you, what's your name?
Orly.
Orlean?
Yeah, Orly. Yeah.
Orly, you get a pathogen and a cell. So because that is the $64,000 question. Why did Ralph, and you can pick whatever pathogen you want. That's a good one. That's chickenpox. So that is the $64,000 question. Why had all these other smart, capable scientists been looking at the very same cell cultures and they didn't see dendritic cells? And that's the magic of discovery. Ralph was special. He was special in a lot of ways. Probably lots of other people saw them and didn't recognize they were different. So I'm going to tell you a story that has nothing to do with immunology, but has everything to do with Ralph's discovery. So have any of you ever heard of Galen? So Galen was the great anatomist of Rome. And Galen, this actually has to do with this, so just bear with me. So Galen was an anatomist who described the insides of people, but he couldn't dissect people. There were very, very strong penalties, a penalty of death for doing dissection or even looking inside people if they were trying to do some sort of procedure. So what he did was he did the next best thing. He dissected monkeys. He dissected old world monkeys because that's what he had available to him. They look pretty much like people, and most of the time he was right. And his anatomic drawings remained the gold standard of anatomy until the Renaissance. So he had a really good run. Very few people's work is considered current and topical for over 1,000 years. But when Galen looked at the heart of an orangutan and he drew it, he drew exactly what he saw, which was 3 chambers. Okay? The heart, a human heart has 4 chambers, 2 ventricles and 2 atria, but an orangutan heart has a common ventricle. So there are 2 atria and a common ventricle. And that's what he drew. But he drew that as a human heart. About 200 years after Galen's death, not only was dissection allowed, but vivisection of prisoners was allowed. As the Roman Empire was decaying, morals decayed along. And so lots of anatomists were looking at human hearts. But until the Renaissance, all of the anatomists drew the human heart as 3-chambered because Galen had drawn it as 3-chambered, and Galen had to be right. So people were looking at a 4-chambered heart, not even something like a cell in a microscope, but something really big and macroscopic before you, and they were describing 3 chambers because that's what they thought they should be seeing. They were seeing what they thought they should be seeing, not what they saw. And in some measure, that's what the other immunologists were doing. They were seeing what they thought they should see, and Ralph saw what was before him. And if you guys go out again with any one message, see what you're seeing. Look and see what's before you and believe your eyes. Because there are probably all kinds of discoveries waiting to be made. And that was one of Ralph's great messages, was there are thousands or millions of discoveries out there. And that's the fun of it. But you have to see what's there and not what you expect to see. And that's why Ralph was able to make the discovery that the rest of them missed. The other thing that allowed him to make it was he believed himself, and he stuck with it when other people told him that he was wrong. I have to confess that I suspect if it had been me and everybody had said, oh no, that's just a funny macrophage, I'd say, oh yeah, probably just a funny macrophage, and I would have moved on to the next thing. But when in that video at the beginning that I showed, where he said, once I saw it, it was simple, that character trait, that it was simple for him, that made it different too. Okay.
I have a question.
Oh, yeah, one's working.
Yeah,
okay.
Does this work?
Sure. Okay, good. So I'll get to you in just a minute. Yes.
I want to know the difference between, let's say, disease germs and other pathogens. What's the difference between how the immune system acts on them?
I'm sorry.
Like different pathogens, right? Like diseases, germs, viruses.
Yep.
How does the immune system go about fixing those problems?
Well, that is a huge topic. How the immune system responds differently to different kinds of pathogens, and how the immune response is made is determined by the dendritic cell. So the interaction of a pathogen, whether it be a virus, a bacteria, a fungus, with the dendritic cell at the very beginning of infection determines what immunologic outcome is going to be. So there are, we would be here till tomorrow if I went through all of the ones that we know about, but in broad strokes, there are some pathogens like smallpox that are terrific for the immune system. They come in, the immune system reacts, and they make lifelong durable immunity, and they clear the virus, and you get better. Then there are some like malaria, HIV, and tuberculosis, where the pathogens have ways of evading immunity, and you get chronic infection. In those cases, you end up with chronic infection, and it's not so easy. The immune response doesn't clear the infection. So those are the two sort of options. And we're going to hear more in a little bit about how the dendritic cell determines those. So for that, you get a pathogen. And what's your question?
If lymphocytes are very specific, this is going back to immunology, then how is it that when a person has leukemia, they get a transplant from somebody else with someone else's lymphocytes?
So that's a great question, but it has to do with the ability of the person who's being the transplant. So when you get a bone marrow transplant, you get a bone marrow transplant because the cells of your immune system, your white cells, are malignant. And so what happens is the doctors give you radiation or chemotherapy to ablate your own bone marrow, to kill it all, and they give you back marrow that's well matched. So you can't just get anybody's marrow. If you got just anybody's marrow, you'd die because your immune response would make a response to the foreign antigens on that other person. So when you hear about there being a drive to get a marrow donor, it's to find a very close match. So the new bone marrow won't stimulate the immune system as being foreign. So the reason that it can work is because you get a very, very close match. And it's not seen as foreign. It's seen as close to as possible as self. And dendritic cells initiate that response as well. And for that, you get a white cell. Next question.
My question is going back to the AIDS vaccine that you mentioned earlier.
Uh-huh.
I was wondering, does that directly protect against AIDS, or does that indirectly protect against HIV?
It protects against HIV infection.
What?
So it protects against infection from HIV.
Yeah. And why does that only work for 6 months?
We don't know, because it's not good enough. We're working on that, because it's not good enough. And for that, we have HIV in here. That's it, HIV. You get HIV. There you go. Okay. Wow. Okay, I'm so glad for all the questions, but I'm afraid we're going to not get through. So if you guys could hold them, I promise I've got enough pathogens and enough bugs, and enough cells, but because we have pizza. Okay. So I have to tell you, I am somebody who comes to work every morning thinking about what I'm having for lunch, and this morning is no exception. So I am thrilled that you're asking so many questions. And each one of you, save the question. I have a pathogen with your name on it, and we'll get to it before the pizza. Okay. So, let's see. So this is what everybody thought was making the system go. This is a macrophage. Okay? But they were wrong, and Ralph was right. So, when Ralph came to work with Dr. Cohen, he started to work on macrophages, because what he thought was that the macrophages, which were picking up, they're phagocytes, they pick up all sorts of material, pathogens and cellular debris, that they were picking it up and then they were presenting it on their surface. But in fact, when colleagues worked, Ralph and Dr. Cohen and colleagues worked on it, it turned out that the macrophages, rather than presenting and showing the antigens to the other cells, they were degrading it. They were breaking it down. They were eating it up into tiny, tiny little pieces. So it seemed like what everybody thought was doing it couldn't be what was doing it. So remember I mentioned Dr. Deduve. So Dr. Deduve and Dr. Pallotti were awarded the Nobel Prize in 1974 for their discoveries concerning the structural and functional organization of the cells. So Dr. Pallotti worked particularly on microscopy and fixation methods, Dr. Deduve on gradients for separating the components of the cell, and he described the lysosome. And Ralph was fortunate because they were here. And this is one of the wonderful things about this institution, and they were able to help him, particularly Dr. Deduve, with developing these gradients. Now this slide was made, as Dr. Krieg told you, my friend and colleague, Michelle Nussensweig, gave the Nobel address for Ralph. And many of these slides were shamelessly stolen by me from Michelle, or actually given to me. But this one particularly is terrific because it shows how the dendritic cells were first made. And whoever asked what the process is, this is the slide for you. So Ralph started with mouse spleens, and you're lucky to just be seeing the spleen and not where it came from. And then they were put over a BSA, or a bovine serum albumin gradient, and then the cells were adhered to glass or plastic. So the cells were put on plastic, and this is how macrophages were made. And so remember, I said we started from the spleen, so the cells were adhered, and then the non-adherent cells were washed off. And in that fraction of the cells were a mixture of primarily B and T cells. And so you were left with these cells. And these here, I don't know how well you can see these stretched out ones are the macrophages. So then they were overnight cultured And some of the macrophages that were stuck stayed stuck. But the cells that didn't stay stuck, the ones that let go and floated up, were the dendritic cells. And that was actually how Ralph saw them. They were the cells floating over the macrophages, if I'm not mistaken. And so then those were taken off, and there were no monoclonal antibodies. So EA rosettes, those are sheep red blood cells, they stick to, it turns out CD1, but to T cells. So the T cells were removed by rosetting. Another albumin gradient was performed. And lo and behold, there you had dendritic cells. After 2 very long days of hard work, you had, like, I don't know, Maggie, how many? 10,000 dendritic cells? Never enough. Dendritic cells were incredibly hard to work with because these are primary cells. So you got a few thousand cells, all of which were post-mitotic and like to die. So the properties that were initially used to distinguish them were the fact that they were non-adherent and poorly phagocytic, where the macrophages were adherent and highly phagocytic. These markers come later. So the dendritic cells are antigen-presenting cells which pick up and then process, and this is really important. So the immature cell picks up the antigen, processes it. As it's processing it, it becomes mature, and then they present it to the lymphocytes in a way that they can be seen, or what's sometimes called in a cognate manner. And this is a picture from Ralph and Dr. Cohen's original description, identification of a novel cell type in peripheral lymphoid organs of mice. There were, I thought there were 3, at least there were 2 articles that appeared in the Journal of Experimental Medicine. And they were reprinted in the November issue. Is that right? In the November issue. So you guys might want to look at that. They're wonderful papers. And Ralph called dendritic cells. He named them for dendrion, or tree. And you can see they're tree-like processes. But that wasn't his first name for them. When Ralph saw them, he wanted to name them for his wife. Some people name cells for themselves, so he could have called them Steinman cells. If it had been the 19th century, that's what he would have done. But that's sort of out of favor now. Not sort of, that is out of favor. But Claudia, Ralph's wife, and I don't know if you mind standing up, Claudia, has very long arms and legs. And so Ralph wanted to call them Claudiocytes, but I suspect that Claudia convinced him otherwise. So that's how they were discovered, and that's how they were named. And this is another one of our beautiful glass dendritic cells from Venice. And so now we're going to see what they do. So his first work in the early 70s was simply, not so simply, but on discovering them. Once he discovered them, then he figured out what they did. And that's when the fun began. This is a newer, I am not. Okay. So this movie was from Michelle, and this is current. This isn't old, but this is how they actually move around. So the yellow cells are the dendritic cells moving in a mouse that's alive. So, did you all see that? Okay, good. Great. So that's what they do. They move around, they stick out their processes, they find antigen, they pick it up, and then they stick out their processes again to embrace the T cells. So this is a dendritic cell from skin. So there are cells in the skin called Langerhans cells. We're going to see in a moment where dendritic cells are in the body. But they're one of the prototypic ones. And they have, when you see them, you see processes, but in 3 dimension, they're really almost these deep ruffles. And the T cells get engulfed and sort of held and hugged by these ruffles.
You have certainly seen thousands like this. They're a real danger. Hi, stop it, you. Stop it. Stop it. Come here. What do you think you're up to? You've probably infected thousands of people already. What do you think this is for?
With every breath, we expose ourselves to hordes of marauding microbes. These invaders are recognized by an army of specialized T cells. But who's commanding this cellular army? The invaders are entering our body all the time. Our dendritic cell, the general, is on a scouting mission. He's hiding at the site of infection. He sneaks in, grabs an invader, digests him, and then displays the enemy's colors. Now he rushes off back to his troops, who are waiting for battle in the immune organ barracks, a nearby lymph node. He shows his troops what their enemy looks like, so they know who to attack. After all, we don't want our soldiers attacking friendly cells. Then, the readied troops multiply like crazy. Now, in battle, they only attack the real enemies, those identified by their dendritic general.
No problem.
So Ralph Steinman discovered dendritic cells, the commanders that rally the troops in the immune system wars.
So, just to review, the cell starts out in the bone marrow, goes into the blood, and then it goes into the peripheral tissue. So that can be anywhere in the body, and we're going to see momentarily where all those places are. And it picks up antigen. And remember, antigen can be anything. It can be a pathogen, it can be pollen, it can be part of your own self. And the magic of dendritic cells are they then process and present the antigen on its surface, and then they go into the afferent blood or the lymphatics, and then they go to the secondary lymphoid tissue, and they present the antigen to T cell lymphocytes. And Ralph and his team did all of the work to elucidate this entire process. So when the dendritic cell and the T cell, so here's the antigen, here's the dendritic cell maturing. When it encounters the T cell, this is sometimes called the immunologic synapse. So there are a whole bunch of receptors that become engaged that tell the T cell, so it sees the antigen in the context of the major histocompatibility locus of the individual. And then the T cell goes out and divides, as you saw in that great cartoon, and that's how you get cell-mediated immunity. The dendritic cells also interact with B cells in not quite the same way, but in a similar manner. So at the beginning, Ralph had a slide that I loved that had the dendritic cells as generals and the T cells and B cells as the soldiers, like you saw. But in the last decade, he thought it was more complicated than that, and he went to this slide, and this is his slide, which is the elegance and scope of immunology, the cells of the immune orchestra. And the dendritic cells were the conductors, and in the beginning, the idea was there was a dendritic cell. And even the first 10 years of my professional life, whenever I got up, I spent the hour trying to convince people that the dendritic cells existed as an entity. But now they're a whole field of immunology, and there are more subsets of dendritic cells, frankly, than I can keep in my head. So I'm not even going to try to teach you those today. Just know that they exist, and they communicate and conduct a whole variety of different effector cells. So the CD4, CD8, and the B cells that we talked about, as well as gamma delta T cells, NK cells, and NKT cells. And the macrophages, the basophils, mast cells, neutrophils, and eosinophils, which are more closely associated with the innate immune system, are playing in the background as well. So, again, a murine splenic dendritic cell from 1973. This is from the original description. And we put another movie here just because they're really fun to watch. So you can see again, understanding that they're picking up, processing, and moving really fast to go find the T cell to which to present antigen. And dendritic cells, in the beginning when Ralph described them in the spleen, that seemed to be the only place they were. And now they've really been found in every place, including the brain. For a long time it was every place but the brain, but in the last couple of years they were described in the brain. They're in virtually every organ, but they are abundant in lymphoid organs like adenoids, tonsils, and lymph nodes. And they form a meshwork through our skin, which are called Langerhans cells, and all of our mucosal surfaces, because those are the places that we encounter antigen. So these are the sites that generate immunity and tolerance. And this is a photomicrograph of the dendritic cells in the airway, and you can see their dendritic processes. And this is a beautiful picture of dendritic cells in the skin. I always like it when they glow green. So again, to review, they come into the lymph node. The dendritic cells come into the lymph node, particularly into what's called the interfollicular or T cell area, and they present antigen to the T cells. They also go into the follicles and present antigen to the B cells. And this is a photograph of a dendritic cell, as I'd like to say, embracing a T cell and presumably presenting antigen to it. And as I said, there are not only many different kinds of dendritic cells, but there are many different kinds of T cells, all of which engage with dendritic cells through different receptors. So here we see, this is just a beautiful picture from our friend and colleague, Nikki Ramani, of the dendritic cells from a human dermal sheet of whole skin. So these are human cells. So remember, I told you up until now, we've been talking about mouse cells, particularly those from the spleen, and now we're going to move to someplace different for the afternoon. So I ran out of images of the beautiful dendritic cells from Venice, so now we have dendritic cells by Henry Matisse, who didn't actually know about dendritic cells. But
we were interested in one particular problem, how the immune response gets started. But really, dendritic cells are turning out to play a role in many other facets of the immune response. Not only does the immune response have to be initiated, but the immune system has to make a choice of what kind of immune response it makes. It is estimated that there are more than 1,000 different kinds of microbes or infections that can attack humans. And the immune system knows how to deal with virtually all of them. There's not one immune response for that 1,000 type of infections. There are many kinds of immune responses, and they have to be selected and tailored in a way that's appropriate to the infection. So the dendritic cell somehow knows how to sense the type of infection and tailor the immune response. But in addition to infection, the immune system has many other important roles that it can play. One of the potential roles that many of us are studying is that the immune system can resist cancer. In cancer, cancers have many strategies to dampen the function of the dendritic cell, so that the dendritic cell presumably is less able to generate an immune response to the cancer. An allergen, substances in pollen, for example, to which many people are allergic, that allergen can affect the dendritic cell so that the dendritic cell then induces what's called an allergic type reaction. In transplantation, when the surgeon puts on a transplant, a liver, a heart, a kidney, dendritic cells from the transplant move into the transplant recipient and teach the recipient that this foreign transplant has been put into place, and that initiates the rejection reaction. And then there are autoimmune diseases where dendritic cells make proteins that drive the inflammation that's seen in disease. So there are many examples of where dendritic cells are exploited during the development.
So in the beginning, Ralph was occupied with showing that dendritic cells started the immune response, the question he came here to ask. But it turns out that dendritic cells do far more than that. So dendritic cells in their classic form begin the immune response. They present antigen to T cells, they cause clonal expansion, and that's how you begin your immune response to whatever antigen. But depending on the type of dendritic cell and depending on the type of T cell with which it interacts, dendritic cells also control tolerance, or our ability to tolerate the cells in our own body, the reason that we don't make an immune response to us. The young person who asked the question about a bone marrow transplant, the reason one can accept a transplant is also due to dendritic cells. And that work has been done in the last decade. So, as I said, we began with dendritic cells from mice, and these are dendritic cells from people. So in the beginning, the cells had to be isolated, and that took days and a lot of mice and a lot of work. And it really limited the amount of discovery that could be done. In the mid-'80s, recombinant cytokines were developed, so synthetic biologics that affect how cells work and grow. And so it became possible then to take precursor cells, so CD34 progenitors from a human being, and treat them with cytokines and make dendritic cells. Teach them to become dendritic cells. So you could grow dendritic cells in culture and make more of them, which is part of the reason that other people began to work on them. Because before that, it was just so hard and so frustrating that not that many people wanted to spend their time doing it. So Theobald Smith was the first director of the scientific advisory board of the Rockefeller Institute, which is what this institution was called at its founding. And he figured out about diseases being caused by vectors. And this is one of Ralph's quotes from him, and I love it. The objectives of research as a mere accumulation of data or the display and parading of acquired knowledge in a world otherwise in motion is outmoded. Discoveries and inventions must be made to yield some contribution towards that rather vague goal, the welfare of mankind. And he said this in 1934. I think it sounds pretty good today, too. So with that said, even though understanding dendritic cells was tremendously important, Ralph was far from satisfied with that, and he extended his work to work on how to manipulate them to make the world better. So the critical experiment that demonstrated that dendritic cells actually are nature's adjuvant and present antigen was done by our friend and colleague Kaio Inaba in the early 90s. And so in mice, she took out dendritic cells from the spleen, from the immune organs, or she made them in vitro, the methodology I described. She added a protein outside the mouse, and then she reinfused them. She gave them back to the mouse. And the mouse was able to fight off whatever, or make a response to this antigen. And that experiment laid the groundwork for the use of dendritic cells in immune therapy, which you're going to hear about this afternoon. So here we have the same idea, Kaio's experiment in a person. So you take out blood cells, you take dendritic cell precursors, you expand them using cytokines, you add an antigen like a vaccine, and then you give them back. And lo and behold, the person makes a really robust immune response. So with that, we're going to break for lunch. It's only 12:03, and time for pizza. I don't know if we should have the rest of the questions now, or maybe hold them we'll hold all the questions for the end. We'll hold the questions for the end. I think we don't want the pizza to get cold. And thank you very much. Well, welcome back. I hope everybody enjoyed lunch. And this afternoon, we're going to move from Ralph's basic discovery of dendritic cells into the medical applications. And it's my pleasure to introduce my friend and colleague, Marina Kasky, who's worked with me and with Ralph for the last 5 years on bringing dendritic cell biology into the clinic. And she's going to tell you about our work with Ralph in that regard. I see you guys all waiting for your questions, and we're going to have all the questions at the end, and I will stay as long as you want. As long as there are questions, we'll stay, and there are lots of bugs left. So let's let Marina talk a little, and then we'll have the questions. But I have to tell you, you guys have made our day by asking questions. So remember what they are. We want to hear them.
Thank you so much for coming back after lunch. So like Sarah mentioned, well, first I want to say that, like Sarah said, my relationship to the lab has been a lot shorter than her relationship. I've only been in the lab for 5 years. So for me, it's really a great honor to be able to be here today and then share with you what I've learned and then the work that we've been doing together. So we learned a lot about the discovery of dendritic cells and what they do. But it was always very important to Ralph to bridge the knowledge, the scientific knowledge in animal experimental models to make a difference in medicine. And like we heard from one of his clips this morning, our immune system interacts with many types of diseases. So we learned that our immune system can fight and resist infections, and it can do the same to cancer. But it can also be involved in rejection to transplants and in the pathogenesis of autoimmune diseases and allergies. And in the middle of all of this are the dendritic cells, since they are the cells that are orchestrating the immune responses. So we believe in the lab that we can, by studying dendritic cells, we can understand disease development, but not only understand how disease evolves, but also use dendritic cells to design new treatments and new vaccines. And our focus now is on developing vaccines. So let's see what Ralph had to say about vaccines.
So let's just think a little about a vaccine. What a vaccine is typically an infectious agent, like the polio virus. And that virus is inactivated or it's attenuated so that it is less virulent, so that it can no longer cause polio. And you then administer that vaccine, typically by an injection under the skin or into a muscle, and the immune system takes that information and develops resistance to the vaccine, in this case the polio example that I brought up, so that when you would be exposed to the real polio virus, you have the capacity to reject it and prevent the body from being infected. But now along comes a situation like the AIDS virus. What we found is that when the AIDS virus interacts with the dendritic cell provides it a home that allows the virus to be transported to the lymphocytes where the virus then replicates. So the evidence says that the dendritic cell is exploited by the AIDS virus to be carried inside the body and to replicate to large numbers. But there's another side to the coin, a second side, which is that dendritic cells can be used to design preventions and therapies for disease. We have been studying how the AIDS virus interacts with dendritic cells, how the AIDS virus uses or exploits the dendritic cells for its own purposes, to move about the body and to replicate. And we're also studying the AIDS virus as the premier example of where we need to use immune principles to design vaccines.
To design vaccines, that's what he said. So what is a vaccine? So vaccines are substances that are used to stimulate the production of antibody. Not only antibodies, but also they imprint a message into T cells. And so that it provides immunity against one or several diseases. It is usually prepared from the causative agents of disease, from the bug that it's causing that infection, or a synthetic substitute. And the origin of the name comes from vaca, from cow, because of the early use of cowpox against the smallpox. So vaccines are among medicine's greatest successes. It is because of vaccines that we no longer have smallpox, for example. But they are still urgently needed to prevent and treat some medical problems, such as AIDS, cancer, and many other infections. So how vaccines came to be. So like Sarah said, it was known since ancient Greece that once someone was sick with an infection, although I don't think they knew there was an infection, but they recovered from that. That person that recovered could then take care of the ill because they could no longer have the infection a second time. So this knowledge was the basis for a concept of this technique that was used in China and India in the 11th century, with the concept called variolation. So the concept behind this technique was to give a very mild form of smallpox in order to provide lifelong immunity. Since we knew that once someone survives an infection, that person can no longer have an infection. So why not give a weak form of that disease so that the immune system can learn and then never get it again? So what they did, without necessarily knowing what caused the smallpox, they collected scabs from. So do you guys know what smallpox was like? So it caused these blisters that then created these pock marks and that scabbed over and then left these scars for life. So they used to collect the scabs and dry the material. And then somehow they realized that it was, the way that it was transmitted was not necessarily from the skin, but it was from the inhaled droplets. So there was virus in the mucosa, the oral mucosa, and then through droplets, the virus then infected the person. So with the material that they collected from people that had recovered from the scabs, they then would, this is a painting showing a person blowing into someone else's nose that material, because somehow they realized that that would then create immunity against smallpox. And this was used for a while. But there were problems with this. The problem was that, for the most part, the disease was a mild disease and people would recover. But there were in some cases, it wasn't quite like that, and then some people died. Also, because they got smallpox, those people that were then variolized, they could then transmit the disease to another person. So it wasn't an ideal vaccination. So years later, Jenner, it came to his attention that milkmaids, even though, say, if you went to a village, you'd see a lot of young ladies with these terrible marks of smallpox. But he noticed that milkmaids they had perfect skins. And he also noticed that even though their skin was perfect, didn't have the pox marks, some of them had blisters on their hands. And then it was known that cattle could have a disease called cowpox. And then probably the milkmaids would be infected with cowpox while they were milking the cows. So he then proposed that perhaps cowpox, infection with cowpox, was protecting people against smallpox. And then he did a key experiment in 1796 where he collected fluid from the blisters of a milkmaid, and then he scraped that material onto the skin of an 8-year-old boy. And then that boy developed little lesions in the skin. And then when he was then challenged sometime later with actual smallpox, he never got sick. So he was able to immunize, to take infectious material from the milkmaid and then vaccinate the boy with that material and then prevent the boy from getting smallpox. And this is where vaccines come from. And then it was a long story ending, for smallpox, it was a long story ending in the late 70s when it was eradicated from the world through vaccination. So, but he did all of that with knowledge that we have an immune system, and then our immune systems, our bodies learn how to fight infection and then can no longer have that infection. However, at that time, there was no knowledge of what the organisms were. So a century later comes Louis Pasteur, where he then initiates what we like to say rational vaccine design. So during his time, one could already isolate organisms and grow them in culture. And then Pasteur was working with cholera in chicken. So he would grow cholera, the bacteria, and then infect chicken. And then he would infect such a high dose of cholera that the chickens would die. So he went on a holiday, and then he left his cell cultures to the care of one of his assistants. But the assistant also went on a holiday. And then when he came back, when they both came back some weeks later, and they used that culture, those cholera germs, to infect the chicken, they got sick, but they didn't die as it was expected. So the assistant said, I think we're better off throwing away these cultures because they're no good. Pasteur, on the other hand, he realized that maybe something very interesting had happened. And there comes that quote where chance favors a prepared mind. So he realized that there was something special. And what he did was he then tried to infect those people with active cholera, and they never got sick again. So what happened was that in those old cultures, the bacteria had become attenuated, it was weakened, and then it caused very mild disease, and then the chicken could never be infected again. So that is the beginning of live attenuated vaccines. And then he went on to make other vaccines, like against rabies, and then other researchers created all these other vaccines over the years. So, but how do vaccines work? So recapping from this morning, we learned that the immune responses are specific. They are directed against a specific pathogen. They are diverse. That's why we can respond to so many different pathogens and microbes that we are constantly exposed to. And then once you build a very strong immune response, your body forever remembers. There is memory, and then you can forever fight that infection. So how does it work? So the vaccine borrows from these properties of immune responses to teach our immune systems to resist infections using the same mechanisms of a natural immune infection, a natural immune response. So the resistance, like in a natural immune response, should be specific for the infection, and then it should typically be durable. And the reason we say typically is because you guys already came up with this, that sometimes for vaccines, we need to have boost vaccination. Because unlike natural infection, sometimes the vaccine does not induce long-lasting immunity. So every so often, we need to boost those immune responses that then wane over time. There are many different types of vaccines. So the first vaccine was a live attenuated vaccine, but vaccines can also be inactivated, meaning the bacteria or the virus is completely killed, but still has the antigens, still has the structures that teach the immune system how to fight it. They can be subunit vaccines, so protein or polysaccharide vaccines. So these are vaccines that have only a particular structure of that bug that the immune system can then recognize and then build an immune response against. And there are more recent experimental type of vaccines, like DNA or viral vector vaccines. So why do we say that vaccines are great successes? So if you look here, this sums up the idea. So here are many of the vaccines that we've received. And this column shows the number of cases that were around in the United States before vaccination was routine for each of these infections. And then many years later, after vaccine was routine, you see that, for example, for diphtheria, it no longer exists. The cases of measles and mumps have dropped. Polio is gone in the United States. And then smallpox is eradicated from the world. So this, to me, really captures why vaccines are so important and why vaccines have been able to help us almost get rid of a lot of these severe infections. However, we still need vaccines for certain important severe and chronic infections, like, for example, tuberculosis, where a third of the world's population is infected with TB. And even though there is a vaccine, it's not such a great vaccine. And it can't really protect against the most common type of tuberculosis, which is pulmonary tuberculosis. In the case of malaria, again, is a very prevalent infection that kills a lot of people. And we still don't have a good vaccine. There are experimental vaccines being tested, but they're still not quite good enough. And then in the case of HIV, over 30 million people are infected, millions of people get infected every year, and then there is some hope that a vaccine can be made, but the best that we have so far is a vaccine that can protect only a third of the people, and like Sarah mentioned this morning, the protection is very short-lived. So it's, again, not ideal. Beyond the scientific challenges, we also have to keep in mind that it's not only having the experiment, making the vaccine. You have to implement. You have to make people believe that vaccines are safe, and then people accept to receive the vaccine. So for example, with polio, we have a vaccine. It's a very effective vaccine. There is an eradication campaign against polio since the late 80s, and still we have polio cases in a few countries in the world, primarily because there are cultural issues preventing people from coming forward and being vaccinated, and then political issues. So we have to keep in mind science, but also society, and then how we can implement science to eradicate disease. So how does a vaccine teach the immune system to resist infections? So captured from what we learned in the morning. So when a person is exposed to an infection or a vaccine, which is a mimic of an infection, the dendritic cells realizes danger entered the body, and then picks it up and then processes and presents on its surface, and then goes to the lymphoid organs to interact with the lymphocytes. And then it finds the lymphocytes that are specific against that infection, and then teaches the lymphocyte how to fight it. And the lymphocytes then go and then can clear the infection. Or the lymphocytes learn how they would fight the infection if it's through a vaccine, so that in the future, if you're ever exposed, the lymphocytes know to go and kill, and you never get sick. So vaccines are a safe way to mimic infection. So again, recapping from the morning, there are different ways to resist infection. There are different parts of the immune response. So the first part of the immune response is the innate immune response. It's what happens right when a foreign organism enters your body. So this response is quick. It's non-specific. So all these cells are just releasing these different cytokines. And it's general. It's not specific for any particular organism. It just happens to everything. But it leads to no memory. But it can kill some microbes early on. Then, a few weeks later, so the innate response sets the stage. It's when the dendritic cell picks up the microbe and then goes to teach the lymphocytes how to fight infection. And a few weeks later, the B cells and the T cells are ready to fight the infection. They already learned their lesson. So then comes along adaptive immunity, which is slower than the innate immunity. It's specific, and then it should last for life. So, and the dendritic cells are in the middle of this. They are really linking this early innate immunity to the adaptive immunity. They capture and sense microbes, and then they initiate and control the type of immune responses that ensue. So when a microbe or a microbe mimic, in the case of vaccine, we encounter a microbe, that leads to an innate response that then matures the dendritic cells. The dendritic cells then travel to the lymphoid organs, where they meet the circulating T and B cells, define the specific ones, they expand, and then they travel back to the area of infection and then try to clear the infection. So now just a few concepts that we've been talking about, but we haven't yet defined. So what is an antigen? An antigen is any foreign or abnormal substance, like a bacteria, a virus, or a tumor cell, that stimulates the body to produce antibodies. So this is a picture of a bacteria, and then here's the antigen. So we can think of the antigen as the building blocks of the different viruses and bacteria. And these patterns are what our bodies recognize and then realize that they have to fight and clear. So antigens are what vaccines are made of, but some vaccines also need to be given together with an adjuvant. And what an adjuvant is an immune booster. So these are substances that are used in combination with a specific antigen to produce a more robust, stronger immune response to the antigen alone. And then these adjuvants are included in a lot of the vaccines that we receive. So now knowing all about how the dendritic cell is key in setting the stage for immunity, and how most of the vaccines are made from attenuated microbes, we asked the question, can we then make new and safe vaccines that are composed only by the antigens, by these defined components that are actually directly on dendritic cells to bring about immune responses? So thinking about that, researchers have come up with two different approaches to using dendritic cells for vaccines. In one approach, which Sarah alluded to this morning, you use dendritic cells from the person. So you generate dendritic cells ex vivo. So what that means is you take a sample of blood, you isolate the dendritic cells, they are expanded in a culture, and then they are loaded with antigens, and then you reinfuse. So in that way, you have a personalized cancer vaccine, for example. But as you can imagine, it's very labor intense, and then it's only done for that one person. So it'd probably be very difficult to bring it to a scale of an HIV vaccine, for example. Another way to think about a dendritic cell is to figure out a way where you can deliver these antigens or these tumor cells directly to the dendritic cell in the body. So you would inject the antigens, and the antigens would find their way to dendritic cells, and then in that way, they would then induce an immune response. So these would be more generalizable vaccines against cancer, infection, and then perhaps other diseases. So this is an illustration for the morning, talking about this first approach. So the person donates blood, the dendritic cell precursors are isolated, then with a mix of cytokines, they're expanded, the antigen is then added to the culture, the dendritic cell matures, processes and presents, and then these cells are reinfused. And then they'll now know how to teach the person's lymphocytes how to fight cancer, for example. And these are some of the labs that are working on this strategy. And in 2010, the first vaccine using this concept of isolating antigen-presenting cells and then adding the antigen in vitro and then reinfusing was approved. So the vaccine is called Provenge, and it was produced by this company called Dendrion. And then it's a similar principle where the patient undergoes leukapheresis, which means their white cells are taken out. The cells are then mixed with antigens against prostate cancer and then an activating substance called GM-CSF. And then 3 or 4 days later, the person is reinfused with those cells. And then they were able to show, and then the reason that they were FDA approved was that this type of vaccination prolonged life of patients with advanced prostate cancer. So this the beginning of the time where we can think that immunotherapy and then perhaps therapies based on dendritic cells can be effective against cancer. But there is a long way to go to make this better. So, but we were not working on this in the lab. We were thinking of this in a different way. So we were using features of dendritic cells to design vaccines. And then we were focusing on 2 concepts. The antigen, we thought that the antigen should be a defined component of a bacteria or of a virus, and they should be directly delivered for dendritic cells. And one way to do so is to target them to these uptake receptors. And I'll explain to you what those are in a little while. These antigens then have to be given together with adjuvants, which are those immune boosters that I told you about. And one type of adjuvant are molecules that signal through these pattern recognition receptors, which are sort of antennas that we have that detect foreign danger signals when we're exposed to them. So together, this is the vaccine concept that we are working on. So but Ralph will probably explain it better. So let's listen to him.
We're learning to direct vaccine proteins to the dendritic cells in the patient. So we don't have to take out the dendritic cells or make them outside of the body. It's much simpler to make a vaccine that knows to go to the dendritic cell and knows to be delivered with other stimuli that the dendritic cell needs to teach the immune system to protect us. And this approach, we think, is not only simpler, but more amenable to translation into global infections and much broader and simpler use.
Okay. So what are those uptake receptors? So we learned that there are many different types of dendritic cells, and then each type expresses receptors on their surfaces. They're slightly different from each other, and there are many of them. And then what these receptors do is they can uptake certain ligands, or they can uptake antigens, and then so that's how things go from the outside to the inside of the dendritic cell, and then they can be processed and presented. And in our lab, we are focusing on this uptake receptor called DEC205, which was discovered both by Ralph and by Michel Nussensweig, one of his very close collaborators. And why do we think that these uptake endocytic receptors are so interesting? Because they interact with microbial self-ligands, so they can enhance antigen uptake and presentation. It's a lot more efficient to bring antigens to a dendritic cell if you bring them through these uptake receptors. But they can also allow dendritic cells to mediate infection. So like we saw in that video earlier on, HIV can hijack a dendritic cell. It can enter through one of these uptake receptors called DC sign, and then it can be incorporated in dendritic cells and then be carried to the other cells of the immune system. So if we have anti-receptor monoclonal antibodies, so antibodies that bind to these receptors, they could then be used to deliver vaccine antigens for improved vaccination. And so that we could also study dendritic cells. So this is the receptor that we are focusing on, is DEC205. It has 10 lectin domains, and it's expressed on cells. Dendritic cells are very abundant in the T cell areas of the lymph nodes, which make this very special because those cells are in very close contact with the T cells, so that when they're presenting antigen, it makes it easier for them to find the specific T cell for that particular antigen. And this is a picture by Maggie, who's here, and Angela, showing so all of this red are DEC205 expressing dendritic cells in the T cell area of the lymph node. And you see that there's some of it too in the B cell areas. So they're really in close contact with the T cells that are circulating through the lymph node, so that they can select which ones are needed to fight that particular challenge. So this is a diagram of what our vaccine is. So we have an antibody, and you learn what an antibody looks like from the morning. So this antibody, it's specific, it binds to one of these uptake receptors, in this case DEC205, and we linked antigens from HIV or from cancer or from other infections, from autoimmunity, diseases like multiple sclerosis. We link to the back of the antibody, if you will, so that when the antibody binds to the receptor, it's taken up and it carries with it the antigen. So then the dendritic cell processes the antigen and then can present to the T cells. But in order for that to take place, the antibody has to be given together with an adjuvant, so that the adjuvant, what it's doing is causing an inflammatory response. So it's telling the dendritic cell, hey, this is not good, you have to fight it. So you have to present the antigen with the alarm signal, so that the dendritic cell knows that it needs to teach the lymphocytes how to fight that particular infection. So the adjuvants that we're using are, like I said, are ligands for these toll-like receptors. And what they are receptors that can detect microbial patterns that are not expressed in humans. And they alert the body of an infection. And they were also awarded with the discovery of these receptors was the second half of this year's Nobel Prize. And I guess the way we put it together is once we are invaded by a microbe, the body detects the presence of the microbe by recognizing these patterns that only bacteria, only viruses have. these receptors alert the cell, that cell being a dendritic cell. Dendritic cell then picks that microbe up, chops it up, and then presents to the rest of the cells. So this is how the innate response is linked to the adaptive response. And there are many different types of TLR receptors that recognize different parts of bacteria. For example, TLR4 recognizes LPS, which is on the wall of gram-negative bacteria. TLR3, which is what we are focusing on, recognizes double-stranded RNA, that it's a pattern in viruses. So we are testing different of these ligands, but so far we've had the most success with ligands for this type of TLR. So just to illustrate what I've been trying to explain, so what is our dendritic cell targeting strategy? So as I said, here's a dendritic cell. They have on their surface DAC205 uptake receptors. So then we give this vaccine that consists of a monoclonal antibody carrying, in this case, HIV antigen. We give this in the skin of the person. That finds the dendritic cells that are in the skin. The dendritic cell then picks up that antibody with its antigen, processes the antigen, and presents on its surface. And then it travels to the lymph node, where it's going to meet the recirculating T cells, and then will meet the specific B cells, and then it'll induce an immune response. So then these lymphocytes will travel back to the periphery, and then will be there, hopefully for a long time, to fight an infection later on if the person is ever exposed to that infection. And then we think, so we are first focusing on HIV, but we think that we could use this vaccine strategy to many other diseases. Not only infections, but also cancer and autoimmune diseases. So our first focus is on HIV. This is a picture of HIV and then its different components. And one of the reasons that we focus on HIV is not only to prove that we can make a dendritic cell-based vaccine, but it's also because HIV, even though we have effective treatments, continues to be a major problem. And it is a major problem, especially in underdeveloped parts of the world, like in Africa. So like I said, even though there is treatment, we can't keep up with it. So for every 100 people that are put on treatment, 250 are newly infected. So we're always falling short. So we really need to focus on prevention. There are many different ways to prevent HIV. None of them are perfect. You all know about barrier methods, condoms, how effective they can be if they are well used. Circumcision has been proven to protect against HIV. And then these other modalities are more recent and they're still being studied, where you could give someone HIV meds either before or after they've been exposed to HIV. But again, this is still under study. It shows promise, but it's still under study. And in the same way, we can say that if we treat HIV-infected people because we bring the load of virus to be so low, that can also decrease the likelihood that that person would then transmit the infection to another person. But as you can see, none of these are perfect. And so really what we need to focus on is on a vaccine, on a highly effective vaccine. But that has been proven to be very hard. And then some of the reasons that it's so hard is because HIV is a very tricky disease, is a very tricky infection. First, no one is ever cured from natural infection, so we can't really learn from the immune system how to make a vaccine. The virus, and one of the reasons is because the virus is very variable. So your immune system, even if it learns to fight virus A, the virus is always changing. So you never know how to fight the new strains that come up every day. The virus also knows how to hide from the immune system. So it integrates into the host genome, and it doesn't divide. It just stays latent. So the immune system doesn't see it. And then to make things worse, it only infects humans. So then it's very, very hard to study it. We have models of in mice and model in non-human primates, but they are not perfect. So all of these together make an HIV vaccine extremely difficult. So we believe that what a vaccine needs to do is needs to induce antibodies. Hopefully they could induce neutralizing antibodies that then would block viral entry. And they should induce CD4 and CD8 T cells, where CD8 T cells would then be able to, if the antibodies failed to block entry and the cell got infected, the CD8 cells could then kill these infected cells. So we want all of this from a vaccine, and it's been proven to be very hard. And those are the T cells helping. So many candidate vaccines have been tested, and the best results to date came from a large study conducted in Thailand, where it was a very complicated vaccine regimen, and then it provided only 30% protection that waned over time. And this is just to illustrate the trial workers in Thailand. And really what I wanted to remember is that it took over 16,000 volunteers to test this vaccine to show that this vaccine did provide some protection. So it was really a major endeavor on the size of the scientists and the volunteers. So what are we doing about it then? So we have our anti-DAC205 antibody, and then we linked to it the HIV GACP24 antigen. And we then give this together with immune boosters so that the dendritic cells know that they have to do something about it. And this is being done in collaboration with a biotech company called Celldex. So this is just to illustrate that dendritic cells are usually in an immature state where they can capture antigens. And then when they see the adjuvant, then they mature, and then they learn that they have to teach the lymphocytes how to fight that infection, how to build an immune response. So we tested this in mice, and then we can tell that in mice that we induce some immune responses. We induce responses that are of high magnitude. We induce responses that recognize different pieces of P24. It's of high quality. The responding cells make many different cytokines. And then in a very restricted experimental model in the mice, we can show that the mice that were immunized could be protected from a virus that expressed HIV. But again, it's all very artificial. So we knew that we needed to then move from the successful experiment in mice to people. And then this is what Ralph had to say about human studies.
I feel that the study of humans is a very important area for discovery, for research. Often we use the word translation to contrast what I'm thinking. We use the word translation to indicate that we know something from a simpler system, like an experimental animal such as a mouse, and we simply have to translate it, apply it to the human condition. But I feel otherwise. I feel we have to study the human condition to discover the relevant scientific principles. So cancer is an example. Cancer is a very difficult disease to model in an experimental animal. So we need to do research in humans with cancer to really learn how the immune system deals with something like a cancer cell. Can the immune system teach the body that a cancer cell should be treated as if it were an infection, and the immune system should attack it? And the potential of the immune system is that it is able to see many changes, and many changes are occurring in the cancer cell at once. So if the cancer cell tries to escape, the immune system is seeing so many of the alterations that it's very hard for the cancer cell to escape. But still, the immune attack, as it is an infection, is highly directed. It's highly specific. It's usually non-toxic. It really targets in on the cancer cell or the infection. And I think this provides the potential for a whole new type of therapy in cancer. But we need research in patients to discover the rules, to discover the principles.
So, but how do we do studies in humans? It's not very easy. These studies are highly regulated by the FDA, and then you have to go through a long process through various phases until a drug or a vaccine is finally approved for use. So, and you might have heard about these phases of human studies. So first you have to go through a phase 1 study, where usually these are studies of about 30, 40 people. And then the focus of this study is you administer a vaccine or you administer a drug to volunteers, to healthy volunteers usually, and then you're looking for safety. First and foremost, you want to make sure that your new discovery is not harmful. It has to be safe. Once you pass that, but you also would like to see a hint that it can be useful. So then you go on to a phase 2 study. And then in the phase 2 study, then you're involving about 200 people or so. And then in the case of vaccine, what you want to show is that you show that it's safe, and now you want to show that you can induce immune responses. So you can measure that the person after receiving that vaccine makes antibody against that particular infection, or has T cells that can fight that infection if ever the person is exposed to it. Once you do that, then to really show that a vaccine has efficacy, that it can really prevent an infection, you have to go on to a phase 3 study that involves thousands of people. And then you saw in the case of the Thai trial for HIV that it had to involve 16,000 people. And you can only imagine how costly this is and then how long it takes. So here in the hospital, we carry on early phase trials. So we do, like Ralph said, we do studies in humans. We try to prove our scientific concepts into humans. And then we conduct these early phase, smaller trials, mostly in healthy volunteers. And one of the studies that we did in our group, I told you that one of the adjuvant that we're focusing on is ligand for TLR3. So it's a double-stranded RNA called poly-IC. So we knew that it was safe from prior studies, but we needed to understand how it activated the immune system, because that would help us design out the trial of our vaccine. So we gave poly-IC to people, and then we studied how people responded. And then we saw that the first and the second day after receiving poly-IC, everyone developed an inflammatory response. So everyone had their arms tender, they had an area of redness. And along with that, when we checked in the blood, we saw that they were upregulating different genes that were involved in fighting infections. And then we compared these genes to a very effective vaccine, to the yellow fever vaccine. And then we saw that both the yellow fever vaccine and the adjuvant were inducing very similar genes. So then that made us believe that this is a good adjuvant, an appropriate adjuvant to alert dendritic cells of a viral infection, or to help the dendritic cell uptake that viral vaccine and then teach the body how to fight a virus. But really our focus is to bring the concept of dendritic cells, of targeting antigens to dendritic cells, to the clinic. So I told you about DEC receptor, and that was discovered in 1995. And then after a series of studies in animals and safety studies in both mice and non-human primates, we're able to, 15 years later, start our phase 1 study of this compound. So this slide is, again, just to illustrate how long things take, and then how you have to stay focused and believe in what you're doing. So we started our study in June of last year, and then it took us about a year or so to screen over 500 people. So 500 people actually stepped forward and then called us willing to volunteer for our study. And then out of these 500, we enrolled 45 people in the 3 arms of the study. And then the study is now ongoing. So it took a lot of work on our side to find these people, but it also takes a lot of commitment from them to participate in the study. So not only they are going to receive 3 doses of the vaccine over a period of 3 months, but they're going to be followed for another year thereafter. And then all these red lines are all the times that they have to come back to the clinic, because we need to both make sure that they don't have too many side effects of the vaccine, but we also want to collect blood so that we can study the immune responses that they are making. So all in all, it takes a partnership between the scientists and then the volunteers, because without the volunteers, we can't do the experiment. And our study is currently ongoing, and then we are blinded as to what the volunteers really received, but we know that they are making antibodies against the vaccine. So I think this is a quote from Ralph, from the series of quotes that Sarah received from him. And then he here defines why he thinks patient-based research is important. So he says, being explicit about patient-based research, it is exasperating in the number of significant obstacles that must be overcome. It's essential for better understanding of disease, development, and treatment. It's equally profound to other spheres of scientific research, and it's exciting. And we agree with him. So now I think I've made the point that vaccines can be very successful. Pasteur was the first one to manipulate microbes to make vaccines. But now we want to do, but his vaccines were always based on the microbe. They're always based on microbiology. Now we want to apply all that we've learned in immunology and the dendritic cells to make vaccines that use the defined antigens and the adjuvants so that we can hopefully be able to prevent infections and other diseases. So in summary for today, I hope that you go home knowing this new cell, the cell that might be new to most of you, called dendritic cells. They have three major features. They serve as sentinels, as they are present in peripheral tissues, so they are guarding our bodies. They can sense when there is an invader, and then they can respond to that by conducting the rest of the immune system to respond, fight, and resist whatever the challenge might be.
Our hope is first to help develop an AIDS vaccine. This has been a long-standing and really challenging problem. One part of the problem is that we don't know how to elicit T-cell immunity. So a second hope I have is that by learning to do this in the setting of an AIDS vaccine, will it be able to extend our research to other vaccine needs? Malaria and tuberculosis, leishmaniasis are all infectious diseases that are in our research strategy. And above all, we also want to bring vaccines to the field of cancer. Cancer immunology is ripe with potential to help patients resist their tumors. But we need to design approaches in humans to test the capacity of the immune system to bring this about.
So that sums it up. So we're starting with HIV, but we are very ambitious. We want to be able to have an effect on many other diseases. Sarah, do you?
So, sorry for the little bit of confusion with the two of us, but we were trying to coordinate. So I'm going to talk at the end about a little bit more about cancer immunotherapy and about Ralph in particular. But I think now would be a good time to take the rest of the questions. So we'll do the questions now, and then we'll do the end at the end. So for all of those of you who had questions, I hope you remembered them, and we will do our best to answer them.
And we still have plush toys.
And we still have plush toys. Okay.
Hi. I have a question about dendritic cells.
Okay.
So
You're in
you
the right
mentioned
place.
on one of your slides that the immature dendritic cells help other lymphocytes know which antigens are self-antigens, right? It helps with tolerance?
Right. So immature dendritic cells, right, establish tolerance or silence. Yes.
Could you describe a little bit about how that works?
Well, dendritic cells, so there are many different types of dendritic cells, and dendritic cells control both arms, both types of immune response. So they both establish immunity as you would have to a virus or to a vaccine, but they also teach the body, teach the lymphocytes in the body what antigens not to respond to. So probably the most, in quantity, the most quantitative antigens to which we're exposed every day are our own dying cells. So it's immature dendritic cells that deliver a signal in concert with the MHC, the major histocompatibility complex, to the T lymphocytes, to a special subset of T lymphocytes that are called regulatory cells that tone down the immune system or induce tolerance. So they do it through what are called Tregs.
So, do they do that while the T cells and B cells are maturing, or like how does that work?
So the way I think of it is, when the dendritic cell encounters something that it's supposed to see and then make the immune system fight it, it sees that in the context of an inflammatory response. So for example, we are being exposed to food all the time, and then anything can be an antigen, but our bodies receive food and in a way that is not inducing an inflammatory response. So it's not telling the dendritic cell, hey, I'm a bad thing, you have to fight me. So it's in that context that when the dendritic cells finds foreign substances without a context of a danger signal, it doesn't mature. It takes it up and it presents, and then it teaches the cells of the immune system, look, this is okay, you don't need to fight it.
Okay. On this side.
How does our immune system recognize whether a pathogen is good or bad?
I think that Dr. Caskey just pretty much answered that question. It has to do, there isn't good or bad, it's whether it's dangerous or not, whether an inflammatory response needs to be mounted. And so that has in large measure to do with what's foreign and what has these, what are called pattern recognition receptors. So there are these special signals that come from viruses, say double-stranded RNA. Viruses have double-stranded RNA. Mammals don't. We only have it in a single-stranded fashion. So double-stranded RNA tells a mammalian body, a human body, that it's dangerous, that it's a pathogen. Bacteria have endotoxin. They have special molecules that signal in that same way, in a related way, through these pattern recognition receptors, or the TLR-like receptors. Okay.
In one of the previous
we're forgetting you're coming down. Come. I'm sorry.
Okay. In one of the previous slides, it said that dendritic cells were in the tonsils.
Mm-hmm.
So if you get your tonsils removed, does that mean that your immune system is weakened because of your loss of dendritic cells?
Well, that's a very good question. Not only are dendritic cells in tonsils, but the tonsils are full of lymphoid tissue. They are almost completely composed of lymphoid tissue. And people have wondered about that. And the dogma, or what people understand to be correct, has gone back and forth. Clearly, people live long and healthy lives without their tonsils, like me. When I was a little girl, in 1963, my tonsils were removed just because I had them. Okay, if you would see articles in the newspaper about whole families being brought in to have their tonsils taken out just because they had them and because the doctors could. We've come to realize that it's probably never a good idea to remove anything in that fashion, and there have been studies that have suggested that there are some complications to having tonsils removed, but I don't believe any of them have ever been borne out. And I guess about 15 or 20 years ago, a series of very rigorous criteria by a group in Pittsburgh, Drs. Bluestone and Stuhl, were developed, which have been generally accepted as to how many infections one has to have to make it worth removing the tonsil. So in some ways, I guess the jury is out, but you're right in that you lose not just dendritic cells, but a lot of immune tissue. But it seems to be that benefit for people who have chronic infections and are sick all the time seems to be worth that risk. Okay.
Thank you.
Come get your white cell or your pathogen.
I was wondering if it's possible.
Can you speak up a little word?
I was wondering if it was possible for T cells to target other T cells if they look like antigens that were specific to that place.
Yes, it is possible for the body to make an immune response to itself. And in some autoimmune disease, T cells can be the subject of that immune response. How exactly that gets turned on, I think, remains a mystery at this point. So you can come down. Next.
I have a question about vaccines.
Great.
So, when vaccines happen and then we develop memory cells, how do the memory cells work? Do they stay active, like looking for the pathogen, or are they dormant until the pathogen comes into our body?
So that's a very good question. The memory cells don't stay active. The memory cells have actually a relatively short life, and they circulate, and then a small number of them become quiescent, and they become resting cells. And they sit there for a very long time, and resting cells can have a very long life, almost the whole lifetime of a mouse, and probably close to the whole lifetime of a person. And only when the pathogen or the antigen comes back, and then you have to mount what's called a memory response or a secondary immune response, which is easier to do. It happens faster and it requires less. And in fact, dendritic cells can be involved in it, but they're not required. Other cells can present antigen. Then the memory cells come out of the bone marrow and very quickly proliferate, and again, you've got your response. And that's the fundamental basis of memory. But the cells have to rest quietly and quiescently for a long time, and can up for a long time in the marrow.
Okay. Thank you.
Come get a white cell.
Hello. Hi. Why are humans able to obtain the virus of HIV, but animals aren't?
Because HIV. So the way that HIV enters the way that HIV infects is by hijacking white cells. And then so to enter the white cell, the HIV virus needs to bind to receptors. In fact, the CD4 cell that defines that type of lymphocyte. So it needs to bind to CD4, it needs to bind to another receptor that on human cells. Beyond that, the machinery inside the cell so that the virus can replicate and then can kill that cell and then infect other cells, only humans have the proper enzymes.
Transcription factors.
Right. So for example, in the mouse, they cannot. The CD4 molecule in the mouse is not the right type for HIV, so it can't really enter the cell. Beyond that, in other animals, if it enters the cell, it cannot replicate. So then it doesn't become a productive infection. But there's viruses that are similar to HIV. For example, there is an FIV for cats, so it can infect cats. So it's a feline immunodeficiency virus. There's SIV, simian immunodeficiency virus, that infects monkeys. So all of these are related, and then all of these viruses are used to teach us how HIV functions, or causes disease.
See if there's a virus in there for her. Okay. Next.
My question has to do with the immune system and chickenpox. I was wondering if it's easier for a child to fight off chickenpox than an adult to shingles, and if you have chickenpox, can you still get shingles?
So let me answer the question backwards. You can only get shingles if you've had chickenpox. So shingles and chickenpox are caused by the same virus, a virus called varicella. And what happens with chickenpox is you get an initial infection and you get a viral syndrome. I guess probably all of you had the chickenpox vaccine, but I can speak with great authority having had natural chickenpox. It's miserable. You get a fever, you feel sick, and you get these itchy pox lesions all over your body, including inside your mouth. And yeah, it's really unpleasant. But your immune system fights it off, and I think we're missing that, but there were some chickenpox in there. Your immune system fights it off, and you recover and get better. But the virus has a special capacity to live in nerve roots or in ganglia. And the immune system keeps it in check. So even though it's there, nothing happens. But then as you get older, if you become relatively immunosuppressed for some reason, you have an intercurrent infection, you get pregnant, or you just have the natural immunosuppression of aging, which happens, that virus can pop up and it travels down the nerve. That's why shingles follows a nerve distribution, because it's being spread along a nerve. So you can only get shingles if you've previously had chickenpox. And the same vaccine, which is given to children to prevent chickenpox is now being given to older people to prevent them, even though they've already had the virus, to keep their immunity to the virus up. So as their natural immunity wanes with age, the vaccine will boost it and prevent them from getting shingles.
Thank you.
Come and let's see if we have a chickenpox there for you. I don't know if there are any left. Yes.
Hi, my question's about vaccines for childhood diseases. How are you able to get a vaccine that's combined for like diphtheria, tetanus, and pertussis? How are you able to get the vaccine all at once?
How are you able to get the vaccine all at once? So that's a terrific question. And when we were sitting down to put these lectures together, Marina mentioned it briefly, but there are all sorts of challenges in making vaccines that are not strictly speaking scientific. And one whole group of them are what's called formulation. And that is, how do you make the stuff? How do you get it into a vial? How do you keep it sterile? And how do you keep it cold? So one of the great breakthroughs in eradicating polio, and I think we have enough time for me to tell this story. So polio vaccine has to be kept cold. There's something called the cold chain. If you think about it, if you need a vaccine to go from here to New Jersey, it's pretty easy. You put it in a box with some dry ice and you go from one freezer to another. But if you're in Cameroon and you're in Yaoundé and you're going out to the bush, there may not be a reliable source of refrigeration where you're going. Transportation may be slow and you may hit a tree across a road. And if the vaccine gets warm, it's no longer useful. So I don't know how many of you had little Hot Wheels cars you painted with hot water and they changed color, or those dolls and you could paint on their faces and they got like lipstick on them with hot water. So that invention has actually saved millions of lives with the polio vaccine. So there are now labels on the polio vaccine that if it goes above a certain temperature, the paint stays warm. So unlike the Hot Wheels that go back, the paint, it's a thermophilic paint. So once it gets warm, it changes color. And so if the cold chain has been maintained, the label will be one color. But if it gets to where it's going and the label has changed color, the people know it's not good and they know not to use it. So before that time, if there was any thought that the cold chain might have been interrupted, the vaccine had to be thrown away because there was a chance it wasn't good. But with this special paint on the labels, now one can confirm whether or not the cold chain has been intact and if the vaccine is good. So who would ever think that that would actually influence vaccine development? So there are all kinds of issues with how to co-formulate the vaccines in terms of the salts that they're dissolved in terms of the volume, so they don't precipitate, so they don't react with each other, that other scientists have devoted their lives to, which frankly are outside the scope of our expertise and outside the scope of this lecture. But it's that area that has allowed the co-formulation of the vaccines. Now there's another question that raises, and that's the issue of immune competition. People have argued that if you give too many vaccines on a given day, that they're going to compete with each other and you're not going to make a good immune response.
So that's basically why you can get a DTP vaccine all at the same time, but you can't get an influenza vaccine and something else at the same time.
Well, that's actually for other reasons, that's medical reasons, but it turns out you probably can get all those vaccines, and your dendritic cells, you've got enough dendritic cells to see them all and orchestrate the response. Mm-hmm. There are other reasons why they don't give the flu, and it has to do with people, just the reaction, the reactogenicity, and people getting sick after the vaccines.
And if you think
Thank
about little kids, when they go to their pediatricians, they receive a lot of shots altogether, and then they can manage.
This is also about vaccines. I was just wondering, like, what it takes to completely eradicate, like, a disease, like you mentioned, smallpox. Like, that vaccine eradicates smallpox, but there's still polio and, like, chickenpox and everything. Like, what would it take to get rid of, like, that disease completely?
So for eradication to occur, the first condition that has to be met is there has to be no non-human host. So the reason that smallpox can be eradicated was eradicated, and the reason that polio can be eradicated is no other animal gets it. So flu, for instance, which is carried by birds and by pigs, with current technology and currently the way we see things, is not susceptible to eradication, because there's no way, we can't vaccinate all the people. There's no way you can vaccinate all the birds. Birds fly in flocks that are wild, and nobody's going to be able to go out throughout the entire world and vaccinate all the birds that can potentially get and carry influenza. But polio is another disease with no non-human reservoir, so that can be eradicated. You have to have a very good vaccine But then, as Marina talked about, it has to do with being able to get to the places where you have to monitor where the diseases are, and then get to the places and vaccinate not only all of the people who have the disease, but all of the people who are susceptible to the disease. So there is a wonderful book. D.H. Henderson was the person who led the eradication of smallpox effort. And he wrote a wonderful memoir about it. And he developed a methodology, again, this is a public health methodology, of ring vaccination. And it's thought that's what allowed the eradication of smallpox. And it's that same strategy that's been employed for polio. Come on down. Next.
Hi. I was just wondering, like, in terms of like allergies, like a food allergy or like a animal sting or like even mosquito bites, like why is there no permanent memory cells that can like block you from getting an infection or like inflammation?
So for allergy, you have memory. Allergy is mediated by a special subtype of antibody. So I didn't go into it, but there are 5 subclasses of antibody. IgG is the most common. There's IgG, IgA, IgM, which is a pentamer, IgD, which is fairly rare, and then IgE. And IgE mediates allergy, and it causes mast cells to release histamine, which is how one causes the allergic reaction. So you do have memory for that. What immunotherapy for allergy, so shots, tried to create, I guess, enough other antibodies of other subclasses that kind of dilute that out. And that also relies on memory.
Is that what like an EpiPen is, like for people?
No, an EpiPen is to treat the actual allergic response. So once the IgE causes the mast cells, do you remember those were all the way on the left? They've got a lot of granules. They have histamine in those granules. The granules release histamine, and then that causes the problems with the airway, and epinephrine, the EpiPen, fights that. And come on down.
For people who get transplants, how do the immunosuppressants sort of decrease the response from dendritic cells?
There are a variety of different kinds of immunosuppressants. Steroids, which are the most general kinds, don't specifically work on dendritic cells. They work more on the T cells and the B cells. There are newer, more specific immunotherapies that are in development, some of which work on dendritic cells. A friend and colleague of ours here named Dana Orange is working on one called tacrolimus, which is, I'm trying to remember the creature from which it's derived. It's derived from a sea creature, and that has a specific effect on T cells and dendritic cells. I think that's an area of ongoing discovery and a good place to work.
I had a question about the pathogens that stay dormant in the body. They can also stay in the brain cells. So does it clog the communication between the dendritic cells and the lymphatic cells? And is there a way to prevent the viruses or to bring them out of their latent stage so the medicine can detect them?
So it's interesting that you asked that. So this is actually something that...
Can you restate what she said, 'cause I couldn't hear?
So she's asking, how can we get a virus out of their latency so that the immune system can then see the virus and then fight and then finally get rid of it, right? So this is something that people talk about in HIV, for example. So with HIV, we have medications, and then they don't get rid of the virus completely, but they keep the virus in check. So you no longer have detectable circulating virus, but there are certainly infected cells. So there are some people that think that perhaps if you take people off therapy, the virus would start again dividing, and then if you then came with a great vaccine or even more potent treatment, you could then eliminate the virus. But this is all still very experimental. And I don't know if we have those tools for other viruses, right?
I'm only familiar with doing it in HIV. But it's an ongoing area to think about how both to get it to come out, how to get it out of the reservoir,
clear,
and then how to clear it and to prevent it from going back into the reservoir. All of those are challenges.
But I think one thing that it's really important to understand is that we have been exposed to a lot of viruses. So probably a lot of us have had EBV, maybe we've had CMV, chickenpox, and we live. So for certain infections, the immune system can completely eradicate the virus, clear the virus, but for others, the immune system keeps the virus in check. So we have the virus there, but it's never allowed to grow out of proportion to cause disease. So that's why, for example, when people that have cancer, they go through immunotherapy, and the immune systems go through chemotherapy. The immune systems are very weakened. So for example, if you had herpes, and then that didn't really cause you trouble for many, many years, once you have chemotherapy and your immune system is so weak, that virus is then allowed to multiply again and then cause disease. So it is interesting to think how we have all these latent viruses that we live with every day.
Can the dormant virus also affect the communication of the dendritic cells with the immune system, or?
Yes, because by being latent, so the cells are infected, but it's not a productive infection, so the cell is not really presenting on its surface the antigen. So the dendritic cell doesn't see it, and therefore cannot kill that infected cell and present to the rest of the immune system. It just stays inside, not seen.
From what I understand, Dr. Steinman had pancreatic cancer, and he did try to treat himself with these dendritic cells. How correct am I in saying that? And did he try to generate these cells ex vivo, or did he try to formulate an adjuvant so that he could deliver it to his cells directly?
We are actually going to talk about that at the very end. So we'll talk about that a little bit at the very end. But there was an article that just appeared. It's in the January issue of Scientific American that goes into that in a fair amount of detail. So I'm going to speak in general terms at the very end. But if you want the more detailed, that's Katie Harmon is the author of the article, and it's available online currently.
Thank you.
You can come down and get a plush toy.
Right. My question concerns the function of the dendritic cells. Now, you talk about how they process and then present the antigens to the lymphocytes, but how do they actually process those antigens? Like, what is different between the antigen that's first presented to that dendritic cell comparatively to that antigen that's presented to the lymphocyte? So what actually occurs in the dendritic cell?
So what, I'm just trying to think where we have a picture of it. So what makes dendritic cells special, among other things, is within the cytoplasm, they have a series of organelles that allow them to pick up at the surface an antigen, say, we could talk about HIV, assuming that it worked for HIV, pick up the whole virus, and then they shuttle the virus through a variety of endosomal compartments that are variously acidified, that break down part of the virus and then re-shuttle up parts of it, and how those parts get determined is, again, part of the magic of dendritic cells, together with molecules from the major histocompatibility complex, which in humans is called HLA, and then are displayed on the surface of the cell. So they goes through a series of vacuoles and endocytic compartments, and the details of that are beyond the scope of this lecture, but it's in several of there are good review articles which we've provided you references to, exactly what the names of the compartments are and the enzymes involved in bringing them apart and then putting them back together with the HLA on the surface.
Thank you.
My question is about the antibodies. Over someone's lifespan, you're exposed to a lot of diseases and pathogens, and your body forms antibodies to fight them off. Does your body store these antibodies, and if so, how and where does it store them? Or does the cells just remember how to make the antibodies and they produce them when they're exposed? Which of these is the correct way?
Right, it's more the latter. So again, like with T cells, the whole trick with B cells is memory. So you get memory B cells, which reside in the bone marrow and rest. And then when the organism, the bacteria, the virus, or the pollen, or whatever it is, comes in again, then they very quickly replicate, divide, and start pumping out antibody. And so the best example of that I know of is yellow fever, So I'm trying to remember the exact dates, but long before people were able to even interrogate the cellular arm of the immune system, there was a wonderful study done in Norfolk, Virginia, with yellow fever. So yellow fever now is a rare tropical disease, but it used to be endemic throughout the eastern United States. And there was a yellow fever epidemic, and I may have the dates of this wrong. I think it's like 1894. And then there was no yellow fever in this town at all until the 1930s. And in the 1930s, there was another epidemic. And what it turned out was that the people who had lived through the epidemic in 1894 mounted an antibody response because they were able to draw blood and detect antibody at that point in 1930, whatever it was. So these were people who had lived a whole lifetime, had maybe not even gotten the disease, but had been exposed to it, made an antibody response, and then a lifetime later, when re-exposed to that same virus, mounted a robust antibody response. And that's because they had resting memory B cells in their bone marrow all that time. It wasn't that they had circulating antibody to yellow fever for their whole life.
But it can be both, though, right? So you can have circulating antibodies, but also have some of the memory stored.
Absolutely, and again, it depends on how much antigen you're exposed to and how much time has passed.
So my question's about allergy medications. I was wondering why the largest side effect that we hear about with antihistamines is drowsiness rather than a compromised immune system. I get that allergies are an overreaction to certain allergens, but why, when you treat them, don't you stop the rest of your immune system from working?
Because an antihistamine is only blocking the histamine molecule, which is the final release product from the mast cell. So it's not interfering with the immune response at all. It's only, so you're making the immune response, you're making IgE, and IgE is triggering the mast cell. The mast cell is releasing the histamine. And so the antihistamine is blocking the action of the antihistamine. So it isn't impairing the immune system. Why antihistamines are sedating, I don't know. Do you know? Sorry, can't help you. But you can still come down.
My question is, what causes the dendrite cells to take its characteristic shape?
Why they have their characteristic shape? That's a very good question, and I don't know the answer to that. I assume it's probably some sort of filaments, some sort of integrins and microfilaments are what give all cells their shape and their structure. But the specifics of it from dendritic cells, I don't know. So you can definitely come down if you have a question we can't answer.
In one of the videos, Dr. Steinman mentioned that your immune system could recognize a cell as a cancer cell because of certain imbalances within the cell. So I'm not exactly sure how a dendritic cell, which recognizes antigens due to uptake receptors, would be able to recognize a cancer cell.
So a cancer cell is not a normal cell. So it has been mutated. So either it has an excess of a certain protein or it has new antigen. It has developed new proteins that are foreign to the body. So that's why then it enters that world of being foreign, and therefore the dendritic cell can recognize and then fight it.
So we have one more plush toy left, which is gangrene, a really good one.
It's Clostridium.
But, that's right. Can you say the name? What?
Clostridium.
It's really called Clostridium. But you might have noticed that most of us are wearing these buttons that Dr. Steinman's family had made, which have an image from the image of the dendritic cell from the first publication in 1973. So they have very generously contributed the buttons for the last few questions because we ran out of plush toys. So.
Hi. So, why did it take so long for dendritic cells to be accepted in the scientific community as its own entity? Like, what was the reason for the skepticism?
That's a very good question. Why did it take so long? And there are several reasons. The first one is scientists are like everybody else, and we like to think we know what we know. And so there was dogma, and there was evidence, and people had their beliefs. So that's the sort of superficial answer. But then there were some other technical reasons. So I guess maybe the more important question to ask is, how does change happen in the scientific community? So somebody has an idea, they publish their idea, they publish their data, and then other people replicate their data. So the most important thing, or one of the most important things in science, is that you can do it and somebody else can do it too. So if I do a set of experiments and I say I've reached conclusion X, and somebody on the other side of the globe who doesn't know me does the same set of experiments or a similar set of experiments and comes to a similar conclusion, then you start to really believe that that there's truth in the result. Because people do experiments all the time that just are wrong, or are what's called an artifact, which is just somehow has something to do with the system and isn't really about the truth. So dendritic cells were tremendously hard to work with. They were, those of us, there are some of us sitting here, not just me, but others, who worked with them in the beginning. They were very rare. There are very few of them. I mean, they're very potent. They are post-mitotic, so they like to die. They don't ever divide. So if you work with T cells or B cells, they divide. So if you start with 100, you have 1,000 in a couple of days. With dendritic cells, you only ever get less. So they're very rare. They're hard to get out. And they like to die. And so it wasn't as though a lot of people wanted to start to try to replicate the experiments because they were so hard to do. So that was one of the issues. So one of the ways they got accepted was when the technologies were developed so they became easier to work with. More people started to work with them because they were conceptually interesting even at the beginning. And I think that had a lot to do with the slow acceptance of them. So I think that those would be the two reasons. I think that's about it. You can come down and get a dendritic cell pin.
In one of the videos, it showed that the HIV virus enters the dendritic cell and it transfers the AIDS virus to all the other cells. So if the vaccine was created, would that mean potentially like destroying some of our dendritic cells?
Yes, it probably would. And probably vaccines that work for other pathogens, for other infections, destroy infected dendritic cells all the time. So one of the things that we didn't really have time to talk about today, and there are hundreds or thousands of them about dendritic cells, is not only do dendritic cells fight viruses, but dendritic cells can also act as the reservoir for viruses. And they act as the reservoir for viruses, for many viruses, including HIV. So there is no doubt that when they become infected, so another virus they act as a reservoir for is a virus called the dengue virus. And that's a virus that people get better from. So there's no doubt when somebody gets infected that some of the dendritic cells that become infected get eliminated. And that would likely be the case if you had a vaccine that worked as well.
But that would have to depend on the vaccine. So I talked a lot about these live attenuated vaccines that have the entire microbe. So those vaccines are able to infect and replicate. For HIV, that wouldn't be safe for the reason that you pointed out, that the HIV would integrate into the cell, and then if that produces a productive infection, then the person would then become HIV infected. So it can't be done for HIV. What can be done is to just get pieces of HIV that cannot assemble and then make it a virus, and then just show to the dendritic cell these pieces that will never become a virus, that will never be able to infect. Okay.
Would it be possible to take antibodies that were made in a lab or from an animal that were specific against the pathogen, put them inside the body and suppress the immune system so they don't attack these new antibodies, and use this to treat different pathogens such as rabies?
That's done. That's called passive immunotherapy. So we've been talking about active vaccination, where you give an agent and you get antibody made by the body. But passive immunotherapy is used fairly commonly for hepatitis B, for rabies, and it's being investigated for HIV. So yes, it's possible and it's being done. Okay, next.
I have two.
I have two questions. My first is related to superbugs. How do superbugs like Staphylococcus aureus resist the immune system, and is that related to the mechanisms of dendritic cells?
So our immune systems are putting a lot of pressure to bacteria. So they're always mutating. So our body learns how to fight one structure of the bacteria, then the bacteria evolves to modify that structure so it can no longer be seen. So that's how by poor use of antibiotics, for example, you kill some bacteria, but you select the ones that are no longer sensitive to that antibiotic. And the same thing can happen to the immune system, for instance. So by modifying their structures, they no longer can be seen. Does that make sense?
Yeah.
Sort of. So that's how we call superbugs. They're superbugs. Some superbugs are really super. They are more virulent. But some of them, for example, for MRSA stands for methicillin-resistant Staph aureus. So it's a Staph aureus that has become resistant to penicillins. So that MRSA is actually less virulent, less powerful than MSSA. But the reason we call it superbug is because not every antibiotic works against it. So sometimes the mutation makes the bacteria weaker, but that can still cause disease because we don't know how to fight it.
Thank you. And my second question concerns stimulating a T-cell response. As I understand it, there are many different types of T-cell response. I've heard that there are some negative effects of a Th1-mediated response. And how does a Th1 response differ from a Th2 response?
So Th1 and Th2 refer to the sort of skewing of the immune response. And in fact, we think of Th2 as being the one that's less desirable for our kind of vaccine. So Th2 is more on the antibody side, and Th1, and it refers to T helper 1 or T helper 2, is more on the cellular immune side. And they're characterized by different cytokines which are associated with them, the manufacturer, but I think, again, that's beyond the scope of what we're discussing today. But I don't think one can be thought of as good or one thought of as bad. It depends on what you're trying to accomplish. They both are important in their own way.
I think one thing that is thought that it's bad is if you have an exaggerated immune response, be Th1 or Th2.
Right. You want balanced.
So you want it to have enough, but not too much. Because if you remember the slide that I showed with poly-IC, people got their arms red. And then if that would have been a little stronger, they could have had fevers because the immune response is induced in secretion of cytokines. And these cytokines can have many effects, including fever. They can lead to drop in blood pressures. So that's why everything has to be in balance.
Thank you very much.
Okay.
Yes, hi. Once a dendritic cell captures a antigen and it shows it to the T cells and the B cells, does the dendritic cell get destroyed or can it be reused?
No, the dendritic cells are not ever reused. That's a very good point, and that's, again, one of the reasons they've been so hard to work with is because they go on and they die typically by something called apoptosis, which is programmed cell death.
So they die once they show it?
Right. Once they do their thing, they're done.
Okay, thank you.
Okay.
My question is there such thing as a defective dendritic cell? And if so, is that diagnosed yet as an illness?
A defective dendritic cell?
Yeah.
Yes, actually, there are defects in dendritic cells. There's a disease called histiocytosis X, and there's a whole group of diseases that are referred to as the histiocytoses. Dendritic cells and macrophages used to be called histiocytes, where there are tumors of these cells, and those have been described for a long time, probably since, I don't know, certainly since the 50s, but maybe since the 30s. But this year, a colleague of ours here, the Rockefeller, Jean-Laurent Casanova, described a primary dendritic cell defect associated, I believe, and I could be mistaken about this, with IL-8 that was published in the New England Journal of Medicine. Yes, yes, they have been described. They're extremely rare, though.
Why are certain diseases found in specific age groups? Does that have to do with the dendritic cell?
why specific diseases are found in specific age groups. I don't think that's specifically related to dendritic cells, at least that I'm aware of. But come get a button anyway. Okay.
Hi. So I wanted to know, I have a few questions. Like, for viruses like HIV that rapidly mutate, when creating a vaccine, if it's different so that it's not as effective as it was the first time, how you were saying how viruses or bacteria become immune to certain defenses in the body. So is it possible that there's a specific target you can use that never changes? It's why you can say, okay, so this virus does this. So if we target that, if the virus were to change, it will be a completely different virus. And then if you want to answer that one, because I have other questions too.
So there, with any virus, but particularly with HIV, there are lots of parts of the viruses that change a lot, but then there are parts that are what's called conserved and change very little. And so we target our vaccines to the conserved portions exactly for the reasons that you mentioned. The vaccine that we're working on is targeted to a highly conserved portion. So it's the same in viruses that can differ up to 30% in some portions. The center portion called GAG that we're working with is highly conserved.
And you talk about how the dendrites teach the immune system how to defend against these things. So does the dendrite teach only one thing, or can they teach multiple things? So when it finds
typically a single dendritic cell presents a single set of antigens, but when you have a population of dendritic cells, they can teach multiple things.
Right.
Okay.
And if you say the dendritic cells die after they do their job, is it safe to say that if you do have a vaccine that may act as a reservoir for HIV or other viruses, wouldn't the cells just die off and the virus also die with the dendritic cells?
I'm sorry, I didn't hear the last part.
Okay, so if the dendritic cells die after taking in the virus or whatever they're looking for, is it after they die, whatever they take in, it's no longer harmful to the body, right?
Right, that's right, except for the fact the problem with that is T cells, which also harbor HIV, are dividing.
All right.
So if the dendritic cells were in isolation, it wouldn't be a problem, but because there are other cells that are dividing rapidly, the virus is being propagated.
All right, thank you.
Okay, come down. Last one over here.
Hi.
Hi.
It's a multi-part question that refers back to an earlier slide. You talked about how now, originally it was a model of the dendritic cell being a commander for the army, but now it's more like the conductor in an orchestra, and that they've actually discovered that there are multiple types of dendritic cells. Could you explain a little bit more about what the significance of the differences between those two models are and the differences between the different dendritic cells, and maybe give a few examples?
Well, I think that the first example is, in the beginning, it was thought that, like the cartoon showed, that dendritic cells talked or instructed only T cells. Ralph's initial work was on the interaction of dendritic cells and T cells. So that was a good analogy for the army. You had the commanders and the soldiers. But it turns out, as we showed you, that the dendritic cells talk and teach not just the T cells, but the B cells, the NK cells, the NKT cells. So that's much more like an orchestra. Okay?
Do you think that there are more discoveries to make about the actual knowledge of what these dendritic cells are, and for example, whether is there anyone trying to see if they can extend the lives of dendritic cells so that they don't just die immediately, or do you think now the significance of Dr. Steinman's discovery is that now it can be used to make all these different immunizations?
Well, I think that both are correct. So the significance is that they can be used, but there's always more to learn. There are so many discoveries to be made. We can't even imagine what they all are. And that's part of the most wonderful, perhaps the most wonderful part of doing this work. You can only imagine so far, and the work goes beyond it. So yes, there are all sorts of, there are countless discoveries to be made about dendritic cells, and you guys are going to be the ones to make them, not us. I mean, maybe we'll make a few more, but it'll be up to you. But I think the importance of the discovery that they can be used stands on its own merit.
Is anyone trying to extend the lives of the dendritic cells, or is that not a focus in research right now?
Not that I'm aware of, but that doesn't mean it's not happening.
Okay.
Okay?
Thank you.
You're welcome. Come down and get a button. Our last two questions.
Viruses change a lot, so does that mean the dendritic cell evolves with the virus, or does it just break it up?
I wouldn't say that the dendritic cell evolves with the virus. The immune response evolves with the virus. So remember, when you think about a dendritic cell, there's a single dendritic cell, and then there are populations of dendritic cells. So any single dendritic cell isn't going to change much with the virus, but the populations will. As the virus changes, the dendritic cells responding to it as a group will change.
But really, the dendritic cell doesn't keep a memory of the virus. So the T cells and the B cells are the ones that need to learn. So the dendritic cells will always detect that foreign material that is coming in. But the T cells and the B cells are the ones that are going to change over time because they're going to be seeing different presented antigens. So they're the ones that are constantly evolving as the virus constantly evolves. So for example, if you get someone with HIV, if that person has ongoing HIV replication, you're going to have groups of T cells that recognize GAG. There are other groups of T cells that recognize envelope. And as envelope changes, you're going to get more and more different types of T cells. But for some reason in HIV, they're always falling behind. The virus is always ahead of them. It's always changing quicker than the immune system can keep up with. But not the dendritic cell necessarily.
All right, our last question.
Yeah, so my question is, if vaccines are designed to boost an immune system response, can we hypothetically use it to boost an old person's immune system and thus live better? Thus live longer? I mean, yeah.
I'm sorry, you have to say, I couldn't hear what you said.
If vaccines are designed to boost an immune response, can we hypothetically use it to boost an old person's immune system and thus live longer?
That's what the shingles vaccine is meant to be. So yes. A lot of old people, they've had chickenpox, and then they've been able to, the varicella virus is still in their bodies, in their neurons, but the immune system is keeping them in check. As you get older, your immune system is weakened, and then some of that virus can then replicate, and then you have what's called shingles. So now, recently, a few years ago, they have a shingles vaccine, and that is exactly the purpose of the vaccine, to give a boost to the immune system so that they can again have more memory cells that can again control the varicella virus.
Okay. All right, well, thank you for all. We have one more question. Okay. All right, this is really the last one.
How did dendritic cells get their names, and do they have any relation to dendrites from their name?
Okay. So I think I mentioned earlier on, dendritic cells got their name because they had tree-like projections. And it's very confusing because there are dendrites in the brain that also have tree-like projections. But other than the fact that they both look like trees, and then they're both cells, that's kind of about it. So they're not related otherwise. Okay. So now, thank you all for your questions. And come get your pen, and thank you. No, we're not done. We're not done. We're not done. Sit back. About 2 minutes and we're done. Okay, we'll go anyway. So the best part is to come. So as was alluded to earlier, at the end of Dr. Steinman's life, he had pancreatic cancer, and he decided to use his dendritic cells to help fight his cancer, which was, I think, what he regarded as the experiment of his life. The details of this are described in pretty good detail in this article that just appeared in Scientific American. So if anybody is interested, I suggest that you go and read the article. This is a picture of a dendritic cell. The work was Ralph's work. but obviously was done by lots of people. And so these are the people who were there at the beginning and through the middle and did the work that you saw. Maggie Whitmer-Pack is here today, and she's also the kind source of all those wonderful plush toys. Michelle Nussensweig is, as was mentioned, gave Ralph's Nobel address, and he's our new lab head. And though he's not here, I think his kids are here. So I think they're probably hiding in the back. I'm not sure that any of the rest of these people are here, but maybe they're here in spirit with us. This is our current lab picture, our most recent one, that was taken about 3 years ago. And I don't know if you can see Ralph. We don't have one of those cool little circle things like we did on the other. But there he is with his lab. This was on the steps of our hospital. And this is all of the people in our lab currently whose work you saw in pieces and parts. This is all of our clinical studies are done here at the Rockefeller University Hospital. Our volunteers. Zach Villa in Public Affairs helped us put the lecture together, and Gloria has run this lovely event for all of us. And as you know, Mary Jean introduced us. But this is the best part, so hold on. Okay. So now I should get Marina up here. So Ralph didn't get to did not get to accept the Nobel, but he did get to accept the Lasker Award. So
ladies and gentlemen, a Lasker Award is exhilarating for the recipient scientist, lab, and institution. In fact, I'm so excited that I'm actually feeling dendritic, wanting to embrace in my processes all the members of the jury and the friends and family who are here today.
So on that note, thank you all for coming, and Happy New Year. Oh, and I forgot the best part. So the future lies with you and other very young scientists. And you met Ralph's granddaughters at the beginning, but this is Ralph's grandson, who's a little young for the lab, but we hold out great hopes. So, Happy New Year.